Hello my dear friends, welcome to Live Prep. Myself Dr. Gajapati Verma. As I informed you earlier, we have started our boot camp. Under this boot camp you will get revision lectures and also test series. And regarding these revision lectures, these lectures are available both on our Live Prep app and also on our YouTube channel. But this test series can be given only through the app. Clear? And regarding this, already I have mentioned earlier also. And now let us quickly go with this revision lectures. In the previous lecture, we studied about the part 1 of the cell, the unit of life. Clear? And there we have studied from the beginning of the chapter until the prokaryotic cells. So now in this lecture we will complete from this eukaryotic cells till the end of the chapter. Clear? Now let us quickly go with the lecture. So here we are talking about the eukaryotic cells. Already we studied about the prokaryotic cells. Let us quickly have some important differences between prokaryotic cells and eukaryotic cells. Now in the case of prokaryotic cells, if we are going from outer side to the inner side, so majority of the prokaryotic cells, they have the cell wall except mycoplasmas. But in the case of eukaryotes, fungi and then protists and some of the protists, not all, fungi, some of the protists and plants, and these are only having the cell wall, but some protists and animal cells, they do not have the cell wall. This is one important difference. Next one is regarding the plasma membrane, how it is compulsory for both. Clear? And next one is regarding cytoplasm, that is also present in both of these. But here in the case of eukaryotic cells, if we are observing, nucleus is present, but nucleus is absent in prokaryotic cells. So since nucleus is present in eukaryotic cells, nucleoplasm is the another word we are adding here. So that concept is not present in prokaryotic cells. And inside the nucleus, DNA is organized into the chromosomes in the case of eukaryotic cells. But in the case of prokaryotic cells, chromosomes are not present. DNA is present directly in cytoplasm, and that is called as nucleoid. Yes or no? That is called as DNA itself is called a nucleoid. Even that location is also called as a nucleoid. Clear? This is what we have studied for the prokaryotic cell. But here in the case of eukaryotes, if we are observing, here there are so many types of cell organelles are present. In the case of prokaryotes, the only type of cell organelles are ribosomes, and they are non-membrane bound structures. Ribosomes are present even in eukaryotic cells also. But along with that, so many other types of organelles are present. Some of them are single membrane bound, some of them are double membrane bound, some are non-membrane bound organelles. So in this way, huge difference are present between eukaryotic and prokaryotic cells. Clear? Now let us quickly go with this eukaryotic cells. So what are the examples under this? Out of the 5 kingdoms, only one kingdom is having prokaryotes, that is the kingdom Monera. What about the remaining 4 kingdoms? They are coming under eukaryotes. They have the eukaryotic cells. What are those? Kingdom Protista, kingdom Fungi, kingdom Plantae, kingdom Animalia. So these are the 4 kingdoms that are having the eukaryotic cells. So that's why these kingdoms are considered as eukaryotes. Clear? Next one. Next one. Here in the NCRT, they gave a sentence that extensive compartmentalization of cytoplasm through the presence of. In the case of eukaryotic cells, we are observing compartmentalization. What is the meaning of this compartmentalization? Let us quickly go through this. Suppose it is eukaryotic cell and this is the nucleus. Okay, forget about the nucleus, they are talking about the cytoplasm, right? Within the cytoplasm again some compartments are there. How the compartments are there? Because membrane bound organelles are present. Some of them are double membrane bound also, some of them are single membrane bound organelles. So due to the presence of a membrane, maybe a single or a double membrane, whatever it is, so due to the presence of these membranes, what is happening? This content inside this is separated from cytoplasm. The content present inside these organelles is separated from surrounding cytoplasm. Since it is separated, it is like a compartment. So that's why please observe carefully. Compartmentalization we are observing due to presence of membrane bound organelles. Membrane bound organelles. How the question can be asked here? So here compartmentalization is due to membrane bound organelles only. Suppose they are saying that in the examination, extensive compartmentalization of cytoplasm in eukaryotic cells is seen through the presence of ribosomes. Suppose they are giving like this. Suppose they are giving like this. Is it true statement or false statement? Simply you may say that organelle. It is not simply about the organelle. You need to have membrane bound organelles. This is the point. So you need to focus on this. Those organelles which are having the membranes, the content present inside them is separated from surrounding cytoplasm. In this way, it is called as compartmentalization. Some students are not giving focus, they are not focusing on some of these words. So that's why I am trying to tell you that these words are also important. So please observe each and every word of NCERT. You don't need to study even a single sentence out of NCERT. Observe each and every word of NCERT, try to understand it, you will score 360 out of 360. Clear? Now let us quickly go to the next one. Regarding the nucleus they are talking. So here regarding nucleus, what they are saying, organized nucleus with nuclear envelope is present in the case of eukaryotes, but it is not present in prokaryotes. That's why this is called as eukaryotic. Eu means what? True. Karyon means nucleus. So true nucleus is present in these cells, but in the case of prokaryotes, true nucleus is not present. Next one here, complex locomotory structures are present. What are these complex locomotory structures? So locomotory, for the locomotion, for the movement of these cells, it is useful. So what are the structures that are useful for movement of the cells? It is about the cilia and flagella. Cilia and flagella are the structures in the eukaryotic cells which are useful for locomotion of the cells, okay. So these are, and why they are saying that they are complex locomotory structures, please observe carefully here. If you are talking about the flagella, flagella are present in both prokaryotes and eukaryotes. But what about the cilia? Cilia are present only in the eukaryotes. Cilia are not present in prokaryotes. Very, very important sentence. Many students are not observing this particular sentence. So observe carefully. Prokaryotes do not have cilia, but both prokaryotes and eukaryotes have flagella. But even though prokaryotes have the flagella, in the previous lecture we studied that prokaryotic flagellum structure is very, very simple. It is not having any microtubules, there is no specific arrangement, and there only flagellin protein is arranged in a coiled fashion, that's it. But in the case of eukaryotes, even though flagellum is present, it is having 9+2 microtubular arrangement, and that is a very complicated structure. Yes or no? And cilia, they are also having 9+2 microtubular structure. So that's why anyhow in this lecture only we will study about those. So that's why in the case of eukaryotic cells, these locomotory structures are complex structures when compared with prokaryotes. So observe this carefully. And then cytoskeletal structures. Please remember, at the level of this class 11th and 12th for NEET UG examination, we are saying that cytoskeletal structures means cytoskeleton is present only in eukaryotic cells. And generally we say that cytoskeleton is absent in prokaryotic cells. We are saying like that. But in higher classes you will study that even in prokaryotes also there is a cytoskeleton. Even in prokaryotic cells also cytoskeleton is present. But again the word complex, you just add it to cytoskeletal structures also. The type of cytoskeleton present in prokaryotes is a different type, and the type of cytoskeleton present in eukaryotes is a different type, okay. So if you can understand this, it is very good, or else at least remember the basic meaning of this particular sentence. Clear? Next one is regarding the genetic material. Regarding this, already I have given the explanation in the previous lecture, the part 1, cell the unit of life, okay. So there we have studied when we need to use the term chromosome. The term chromosome is used only for eukaryotic cells. It should not be used for prokaryotic cells. What is the reason? When we need to say the chromosome, when DNA is associated with histone proteins, then only it will form the chromosomes. So that's why DNA is associated with histones and organized into chromosomes. Where it is applicable? It is applicable only for eukaryotic cells. It is not applicable to prokaryotic cells. Explanation I have already given in the previous lecture. Okay, next one. So major differences between plant and animal cells. So with respect to some of the components, we can find the differences with respect to the cell wall. So with respect to cell wall, regarding the plant cell, if you are observing, plant cells are having the cell wall, whereas animal cells, they do not have the cell wall. Next one is regarding the vacuole. What type of vacuole? Here the vacuole means it is about the sap vacuole. So sap vacuole is present only in the plant cells. Sap vacuoles are not present in animal cells. Please remember, here also I am saying another point. Generally in the animal cells we say that vacuoles are absent. There the meaning of vacuoles is sap vacuole, please remember. But there are different other types of vacuoles. So in higher classes you will study that even in the animal cells also some other types of vacuoles are present. Clear? So please remember this. So here the vacuole means sap vacuole. So sap vacuoles are present in plant cells, not in animal cells. Clear? Next one, regarding the plastids. Plastids are also present in the plant cells, but they are not present in animal cells. But regarding the centrosome, so in the general comparison what we say, centrosome is present in animal cells, but it is not present in plant cells. Clear? But if you are clearly observing the NCERT sentence, they said that centrosome is present in animal cells, but it is absent in almost all plant cells. Please observe carefully. Centrosome is absent in almost all plant cells. Why they are saying almost all plant cells? Why they are not saying all plant cells? This is also another important point. Why they are saying almost all? Because lower plants like bryophytes and pteridophytes, please remember, once again I am repeating, lower plants like bryophytes and pteridophytes, they have centrosome. One centrosome has two centrioles. So bryophytes, pteridophytes, they have centrosomes. So cell division in the case of bryophytes and pteridophytes, so in that cell division process, once again, centrosome means centrioles are involving like animal cells only. Then now the question is, then why you are saying that centrosomes are absent in plant cells? For the general comparison, we are not talking about the lower plants. For the general comparison, we are talking about the higher plants like angiosperms. If you are talking about angiosperms and the majority of the gymnosperms, they don't have centrosomes. So that's why in the general comparison, we say that plant cells do not contain centrosome, animal cells contain centrosome. In this way, we are saying, but where they are present, where they are not present, this point also you need to observe very carefully. Clear? So these are some important points. Next one is regarding the microvilli. So actually in the animal cell diagram, so this is the plasma membrane. Plasma membrane is having finger-like projections. The finger-like projections present on free surface of some of the animal cells are called as microvilli. Clear? And this microvilli was mentioned in the labeling, but they have not given any sentence in this particular chapter. Clear? So please observe this one also. So here regarding the microvilli, in the previous lecture I told you, mesosomes present in the prokaryotic cells, chromatophores present in the prokaryotic cells, and cristae of eukaryotic mitochondria, and microvilli of animal cell plasma membranes. All these are foldings. These are all foldings. Please remember, foldings will increase surface area. So what is the major function of this microvilli? Microvilli will increase the surface area, so that according to the location and function, some are involving in more and more absorption of the nutrients or some other things, and some are involving in more and more secretion, maybe depending on the condition. Clear? So this is some basic information that we need to remember. Next one, regarding the cell wall. And another point, so at the end of this particular lecture, we will talk about microbodies. Under the microbodies, we will talk about the peroxisome. So please remember, peroxisome name was mentioned in the animal cell, and even in the plant cell also they have mentioned this peroxisome. So it is present in both. Peroxisomes are present in both, so please remember this. And regarding the peroxisome, they have not given any content in the NCERT. But since they have at least mentioned in the labeling, at the end of this particular chapter, when we talk about the microbodies, there at least one or two sentences we will study. Clear? Now let us continue further regarding the cell wall. So here what are the cells that are having the cell wall? Plant cells are having the cell wall, fungal cells have the cell wall, and some protists have the cell wall. And animal cells do not have cell wall, and some protists do not have the cell wall. So this is what the basic information regarding the cell wall. So if cell wall is present, what is the nature of this cell wall? Cell wall is a rigid structure. Under the prokaryotic cell wall also I told this. So it is a rigid structure and it is a non-living dead structure. Very important point. So cell wall is non-living and rigid structure. If you are talking about cell membrane or plasma membrane, what you will say? Plasma membrane is living structure and plasma membrane is semi-solid or semi-fluid or quasi-fluid structure. Please understand this. Once again I will repeat this because this is very important one. Cell wall is non-living, means dead structure, but plasma membrane is living structure. Cell wall is rigid structure, that's why it is giving shape, that's why it is giving protection, yes or no? But plasma membrane is semi-fluid, quasi-fluid or semi-solid structure. So these are some important points that you need to remember. Next one, cell wall. So what is the location of this one? It is the outer covering for plasma membrane. It is present on outside the plasma membrane. And where we are observing? In the fungi, plants, and some protists, not all protists. Some protists have this cell wall. And what are the functions of this? In the NCERT very clearly they mention this. So since it is a rigid structure, it will provide the shape, and it is protecting the cell from the mechanical damage, and it is protecting the cell from the infection, and it is also helping in cell to cell interaction, because so if they are multicellular, what happens? So cells are present together. Yes or no? In these cases, fungi and plant cells. So if they are multicellular, cells are attached to each other. Now what is the layer that is attaching to adjacent cells? It is again the cell wall only. So it is helping in cell to cell interaction. And next one, it is acting as a barrier to undesirable macromolecules. So here for this point, you need explanation, and this explanation I will link up now itself. And what is the question here? Cell wall is, what is the nature of cell wall? What type of permeability it is having? It is a freely permeable. Why it is a freely permeable? Understand this. So why it is, how it is acting as barrier to macromolecules? Macro means large molecules, that point. And why it is freely permeable? And here, and then what is the nature of this? It is a porous structure. I will tell you about that particular point. Observe carefully. So in the case of cell wall, so many layers are present like this. Many layers are present, and these layers are again, they are cross-linked. So in different organisms, means in different types of organisms, so different components are present, but overall it will be like this. So overall, if you see, this is like a mesh-like structure. It is a mesh-like structure. So when it is making a mesh-like structure, you can find some pores. You can find some gaps. Technically, they are called as pores. So that's why we say that cell wall is a porous structure. Cell wall is a porous structure, pore-like structure. So in this way, some gaps are present in this. So now how you are interlinking this one? So please observe this one. Suppose there is a molecule. Suppose there is a molecule that is having a larger size. Suppose this is a molecule that is having the large size. So can this large size molecule pass through this small pore? No, it cannot pass through. So that's why please remember, so this is a large molecules, means macromolecule. So how cell wall acts as a barrier for macromolecules? Since it is a porous structure, so it is having a particular size of the pore. So if this is the size of the pore, can large molecule pass through this? It cannot pass through this. In this way, cell wall acts as a barrier to the large molecules. Clear? Next one. How it is acting as a freely permeable? Please remember, it is not having any specificity. So this is the pore size. Any molecule that is a smaller than, suppose this is a small molecule. Any molecule, this molecule is smaller than this pore. So any molecule which is smaller than the size of this pore can easily pass through this pore. That's it. So in this way, suppose these are the pores. Now suppose this is a small molecule, it can easily pass through this because the size of the molecule is smaller than size of this particular pore. So in this way, so small molecules can freely pass through this. That's why it is a freely permeable. And large molecules cannot pass through small pores. That's why it is barrier to macromolecules. Both the points you can interlink. Clear? So this is what they are saying. Next one. Now individually in each type of organism, so what are the various components that are present in the cell wall? So if you are observing the cell wall of the fungus, please remember, don't think that only chitin is there. Some students are thinking that only chitin is there. Some students are thinking that plant cell wall is having only cellulose. It is not like this. Generally we are saying about the major component. So fungal cell wall is mainly having the chitin. Along with the chitin, anyhow this information is not needed for you. Along with the chitin, there are many other components also. Major component, majority of the proportion is by chitin. So this is what the information is. And regarding this chitin, you should compulsorily remember, because in biomolecules you are studying, and also you are studying under the biotechnology chapter. Yes or no? So cell wall of fungus is made up of chitin. What is the biochemical nature of this chitin? This chitin is a polysaccharide. And what type of polysaccharide? Homopolysaccharide or heteropolysaccharide? It is homopolysaccharide. And when it is homopolysaccharide, what is the meaning of homopolysaccharide? One single unit is repeated for several times. So what is the name of that single unit? It is N-acetyl glucosamine. Yes or no? N-acetyl glucosamine is repeated for several number of times, so that it is forming a homopolysaccharide called as a chitin. And this chitin is a polysaccharide. So please remember, that means it is a carbohydrate. Clear? And this is the major component of fungal cell wall. And next one is regarding the algae. So algae and higher plants separately we are studying, but whatever it is, major component will be cellulose only. But along with the cellulose, what are the other components? So that also they have mentioned in NCERT. In the case of cell wall of algae, along with the cellulose, galactans, cellulose. First we will talk about the cellulose. What is cellulose? It is also a polysaccharide. It is also a homopolysaccharide. So homopolysaccharide means, in this case, what is the repeating unit? It is the glucose. Cellulose is a homopolysaccharide of glucose. So glucose is repeated for several number of times. Anyhow, more details we will study in biomolecules. And along with this, galactans, these are also polymers and mainly having the galactose. Mannans are also polysaccharides, mainly having the mannose sugar. And then along with these carbohydrates, it is also having some minerals like calcium carbonate in the cell wall. So this is what about the algal cell wall. And now our main focus is on higher plant cell wall. So in the case of higher plant cell wall, cellulose is present. Anyhow, it is a homopolysaccharide made up of glucose. And along with this hemicellulose, it is heteropolysaccharide. Different components are present. Pectins are present. Here also it is heteropolysaccharide. Here also different units are present. And along with this, proteins. This is not a carbohydrate. Proteins are a separate group, right? So proteins are polymers of amino acids. So in this way, different components are present in the cell wall of higher plants. This is what the basic information that they are saying. And if we are observing the layers, so before directly going with the layers, first observe this particular labeling. In this labeling, at the center, they have mentioned it as the lumen. And regarding the lumen, in the previous lecture, I told you, when you will use the term lumen, if it is empty space, please remember, this is a cell, right? So when we studied about the Robert Hooke, at that time I told you, at the center, empty space is there. That empty space for this particular dead cell, for the dead cell only you are observing the empty space. The empty space present at the center of this particular dead cell is called as a lumen. And why it is empty? Because the cell is dead. That protoplast, what is protoplast? Plasma membrane plus protoplasm, it got dried up. Since it got dried up, now you are observing empty space. This empty space is called as lumen. Suppose it is a living cell. Now here inside this you can see plasma membrane, and inside that you can see protoplasm. Yes or no? This is what the point is. Concept, words, meanings, everything you need to understand. Clear. Now let us talk about this particular cell wall. For this cell wall, what is the outermost layer? This is for one cell we are talking. Outermost will be middle lamellum. Below this means what? Towards the plasma membrane side. Below this will be primary cell wall, and below that will be secondary cell wall. Generally this secondary wall will be having different layers like S1 means secondary layer 1, secondary layer 2, S1, S2, S3, likewise different layers are present. And in some cases, very rare cases, you can find the tertiary wall also. In very rare cases you are observing the tertiary wall, but in the NCERT they have not mentioned about the tertiary wall. So we are talking only about the primary wall and secondary wall, that's it. Okay, and after this will be the lumen for the dead cell. If it is a living cell, after the secondary wall, what you will find? Plasma membrane, and inner to that protoplasm will be there. This is what the sequence. Clear. Now what we are saying, suppose this is one cell, now here is the other cell. Suppose this is one cell, here is the other cell. So now two adjacent plant cells, they are connected by what? What is this one? It is the middle lamellum. So middle lamellum is the layer of cell wall that is connecting adjacent cells. That's why it helps as a glue or a connector for the adjacent plant cells. This is some basic information that they are talking. Now let us go with the more details. Middle lamellum. So middle lamellum is mainly composed of calcium pectate, and its function is it holds or glues different neighboring cells together, because two adjacent plant cells are connected by middle lamella. And another point that they have mentioned in the cell cycle cell division chapter is, actually, in the plant cell, during the cytokinesis, a cell plate is formed at the center. Cell plate is formed at the center, and this cell plate will slowly mature due to deposition of some components, and finally this cell plate will be converted into middle lamella. Middle lamella. Okay, so this is what the name that they are using here. Next one is regarding the primary wall. What is the location of primary wall? It is present just inner to, suppose this is the middle lamella, just inside this. This is the plasma membrane, right? Towards the plasma membrane side. Just inner to the middle lamellum is the primary wall. And this, if you are clearly observing the NCERT, they said that primary wall is the cell wall of young plant cells. Please understand the logic there. Cell wall of young plant cells is called as a primary wall. Why you are calling primary wall as cell wall of young plant cells? Why you are not calling this as cell wall of mature plant cells? You need to understand these words. Then only you can understand NCERT words. Okay. Why it is called as cell wall of young plant cells? Because young plant cells, they do not have a secondary wall. Young plant cells, they do not have secondary wall. Please observe this. Young and growing cells, they don't have secondary wall. In those cells, primary wall only is present. That's why it is called as cell wall of young plant cells. Please understand this and how it will grow. And here you just observe this. There is a word called as intersusception. Primary wall will increase in length by intersusception. What happens in intersusception? You need to understand two words. One is intersusception for primary wall, opposition for secondary wall. Now let us observe carefully here. Suppose this is a small plant cell. Suppose this is a small plant cell having some particular thickness. Okay, and this is a growing cell. If it is a growing cell, it will not have secondary wall, and it is having the primary wall. Outside this, middle lamella will be there. But our point is how this primary wall will increase in size. Observe carefully here. Cell wall materials are synthesized. Cell wall materials are coming from the Golgi complex, and on the plasma membrane these cell wall materials are attached to each other. From the plasma membrane they are targeted to the cell wall. Now they are coming to the cell wall. Please observe carefully. When they are coming to the cell wall, now what happens is this primary wall thickness will remain same only. But what happens is the material will be added in between. Materials are added in between. Once materials are added in between, then what happens? Overall length of this particular primary wall will increase. Please understand this. Thickness remains same. Thickness remains same, but the length will increase. This type of growth is called as intersusception. This is what the point we are writing here. Okay, and here this primary wall gradually diminishes. What is the meaning of diminishes? Decrease in thickness. Already it is thin and it will further becomes very thin. It becomes further thin as the cell matures. Why it is becoming further thin? Because of the cell maturation. Because when the cell undergoes maturation, then secondary wall formation will occur. And how secondary wall will be formed here? How secondary wall will grow here? Suppose this is primary wall. Suppose this is the primary wall. Cell has reached its complete growth. Now it is undergoing the maturation. During maturation, what happens? Inside this, secondary wall will be formed. Secondary wall will be formed. But please remember, secondary wall length will not increase. Thickness will increase. For the secondary wall, thickness will increase. Please observe carefully here. For the secondary wall, thickness is increasing. So materials from plasma membrane are coming. So here they are deposited layer by layer. Layer by layer they are deposited. So thickness for the secondary wall is increasing. So this type of growth is called as apposition. Please observe carefully. This type of growth is called as apposition. So secondary wall will grow by apposition, whereas primary wall will grow by intersusception. These are two important points that you need to understand. And the secondary wall is formed inner to the primary wall. Clear? Next one regarding the plasmodesmata. So plasmodesmata are the direct connections between the plant cells. So here what exactly happens? Let me draw one rough diagram. Suppose this is the middle lamella. I am drawing only one plasmodesmata. Suppose these are the two adjacent cells. So what are the plasmodesmata? Originally actually it is the normal cell wall, but plasmodesmata are some gaps that are present within the plant cell wall. And how exactly these gaps are present? Suppose this is the middle lamella, okay, and inner to this is the primary wall. Inner to this is the primary wall, okay, and inner to this is the secondary wall. Secondary wall up to this, okay. So this is how it is present. This is how it is present. So in this way, there is a complete gap here. It is like a pore, okay. And now what is the important feature that we need to understand here? The point we need to understand is plasma membrane of one cell. This is the plasma membrane of one cell. Plasma membrane of one cell is continuous with plasma membrane of adjacent cell. Understand this. Plasma membrane of this cell and this cell are directly connected. So that's why. And now this particular one, this particular pore is called as a plasmodesmata. At the plasmodesmata, what is the membrane that is present here? Plasma membrane. Plasmodesmata are covered by plasma membrane. So now another important point we need to understand is, since plasma membranes are continuous, now cytoplasm of one cell is continuous with cytoplasm of adjacent cell. And not only this, suppose this is the nucleus of one cell, nucleus of other cell. Now this is the endoplasmic reticulum of one cell and this is the endoplasmic reticulum of other cell. So endoplasmic reticulum of one cell is continuous with endoplasmic reticulum of other cell also. And here this is the endoplasmic reticulum is going like a tube through the plasmodesmata. So that's why it is called as a desmotubule. So in this way, huge information is there. But anyhow, this much of information was not given in NCERT. Simply remember that plasmodesmata connect cytoplasms of adjacent cells. So that's why plasmodesmata can form simplest. Okay. So this is what some important information, and the advantage with this plasmodesmata is quick information transfer, ions transfer. Some small molecules can also be easily be transferred from one cell to the other plant cell. This is what the advantage of this. So plasmodesmata connect cytoplasms of adjacent cells. Clear. Next one. So with that, regarding the cell wall, it is completed. Now we are going with the cell membrane or plasma membrane. And here, if you are clearly observing the NCERT, they said that especially human RBC are used for the study of plasma membrane or cell membrane. Now you need to tell me the reason. What is the reason behind the use of human RBC for plasma membrane studies? So whenever I ask this question, immediately students say that human RBC, mature human RBC does not contain nucleus. That's what they are saying. But please remember, okay, that is correct to some extent. It is not just the absence of nucleus. Please understand this point. It is not just the absence of nucleus. All the cell organelles, all the membrane bound organelles will be lost. Please remember, not only nucleus, other membrane bound organelles are also absent. This is what the important point is. Now, what is the logic of using this particular one, and what is the relation with this particular concept? Observe carefully. Suppose this is the human RBC. For the human RBC means it is the animal cell. It is the animal cell. For the animal cell, outermost layer will be plasma membrane. Yes or no? And what is our aim? Our aim is to study the plasma membrane. Suppose you are taking some other cells, other animal cells, then what happens? Plasma membrane is present and other membrane bound organelles are also present. Yes or no? So in this way, what happens is, suppose I am a scientist and I wanted to isolate this particular membrane. So whenever I am trying to isolate the plasma membrane, there is a chance that membrane of the organelles also coming along with the plasma membrane. There is a chance. So now what happens? You cannot get the exact composition of that, and you may get some misleading results. So this is what the problem with other cells. But if you use human mature RBC, what is the advantage? It does not contain, anyhow nucleus is not present. Not only nucleus, other membrane bound organelles are also not present. Since other membrane bound organelles are not present, if I am using this particular human RBC for my experiments, then the only membrane that I am getting will be plasma membrane only. There is no mixing, there is no contamination. The technical word is contamination. There is no contamination with membranes of the organelles. So that is the reason why we are using human RBC especially for the studies of plasma membrane. This is what the reason they gave. Please remember all these things. They may ask in a certain reason things. Okay. Next one is, so what actually it is composed of? Four major things that we are studying. Plasma membrane mainly composed of four things. Phospholipids are present, and then proteins are present, carbohydrates are present, and cholesterol is present. These are the four important things. So one by one we will go through. Out of these four, again, anyhow, out of these four, what are the major things again? Lipids and proteins are the major things. So first we will go with this, and then slowly one after the other we will go through. So cell membrane is mainly composed of, even though these are the four things we are studying, out of this again mainly lipids and proteins. And in the lipids also, different types of lipids we have studied in the biomolecules. But in the case of membranes, what type of lipids are more present? Phospholipids are present. In the phospholipids also, what type of phospholipids are present? Now these are diglyceride type of phospholipids. That means to the glycerol, to the two carbons, two fatty acids are attached. To the third carbon, phosphate group is attached, and to this phosphate group again some other groups may be attached, so that they are further substituted. So this is what the basic structure of this phospholipids. And how these phospholipids are generally arranged, monolayer or bilayer? Generally they are arranged as bilayer. This is how they are present. Monolayer means single layer of phospholipids, but generally in the membranes we are observing phospholipid bilayer. Bi means two layer. This is one layer, this is another layer. So please remember, so two layers combinedly they will form one membrane. So if you are saying about plasma membrane, it is one membrane. That means 2 layers of phospholipids. So in the same way, this is the basic arrangement even for membranes of organelles also. If you are talking about endoplasmic reticulum, Golgi complex, mitochondria, there also membranes are there. There also membranes are there. They may be single membrane or double membrane, but there also membranes are having bilayers only. So endoplasmic reticulum, Golgi complex, so lysosomes, vacuoles, if you take these examples, these are all single membrane bound. Even the microbodies also, these are all single membrane bound. Single membrane, one membrane means 2 layers of phospholipids. If you are talking about mitochondria, chloroplast, nucleus, they are double membrane bound. Double membrane bound means this is one membrane, like this another membrane is there. So total 2 membranes are there. 2 membranes means 4 layers of phospholipids. So when you are reading the question, please read the question carefully, whether they are asking about membrane or layers of phospholipids. One membrane, 2 layers of phospholipids. Two membranes, 4 layers of phospholipids. So read the question very carefully. So this is how generally phospholipids are present as bilayer. If it is single membrane, so it is bilayer, means only 2 layers are there. But if it is double membrane, in this membrane 2 layers, in this membrane again 2 layers. So overall 4 layers of phospholipids are present. So please read the question very carefully. Clear? This is what next one. So how exactly they are arranged? So in the NCERT very clearly they said that hydrophilic head groups are present on the outer sides, whereas the tails are, they are hydrophobic or they are non-polar. Please remember, hydrophilic heads are polar heads. They are present on outer sides of the membrane so that they can freely interact with the water, whereas the tails are non-polar or they are hydrocarbons, saturated hydrocarbons mainly, not always, mostly they are saturated hydrocarbons, and they are present on inner side of the membrane so that they are forming the core. Central core of the membrane is made by these hydrophobic tails. Okay, so since they are hydrophobic, so since they are present on inner side of the membrane, so that they are present away from the water, so that this structure will give the most stability for the membrane. That's why they are present as bilayer. Clear? This is another basic information that they are talking here. Why non-polar tails of saturated hydrocarbons are present on inner side of the membrane? This is the assertion reason type. They are present on inner side. What is the reason? So they are hydrophobic in nature. So if they are present on inner side, they can stay away from the water. They can avoid contact with the water. So that they can give more stability to the membrane. That is what the reason is. So assertion reason wise, like that also you need to see. Next one is regarding the cholesterol. So is cholesterol present in the membranes? Yes, it is present. So in the case of animal cells, if you are observing on both the layers, whether it is upper or bottom, wherever it is, so cholesterol is present between these phospholipids. So it is interacting with the phospholipids. So ultimately it is useful for maintaining the membrane fluidity. So that membrane should be, this is plasma membrane is quasi-fluid, right? Semi-fluid in nature. That membrane fluidity maintained in different conditions like cold conditions, hot conditions, normal conditions. So in all these conditions, the membrane fluidity is maintained with the help of this cholesterol presence. Another point is, in the case of animal cells, cholesterol is present. In the case of plant cells, phytosterols are present. In the case of fungal cells, ergosterols are present in general. So please remember this. Next one is regarding the components. What are the other components? Lipids part we have completed. Proteins are also present. Cholesterol, carbohydrates, all these are present. And now we will see what is the location of the carbohydrates in the plasma membrane. So if carbohydrates are present in the plasma membrane, this is the NCERT diagram, right? For the NCERT diagram, this is exoplasmic side, outside the cell. This side is towards the cytoplasm. That's why it is called a cytoplasmic phase. This phase of the membrane, in this diagram, the bottom side is facing towards the cytoplasm. So that's why it is called a cytoplasmic phase. This side is present outside the cell. So that's why it is called as exoplasmic phase. So first you need to know the names, then only you can answer my question. So in this diagram, you just observe and you just tell me on which side the carbohydrates were mentioned. Only on exoplasmic side, only on cytoplasmic side, both the sides, and no side. So observe the diagram carefully and tell me the answer. On which side they have mentioned? Sugar means carbohydrate, right? So they have mentioned sugars only on exoplasmic phase. This is very important point you need to understand. Sugars, if they are present in the membrane, they are present only on exoplasmic phase. They are never present on cytoplasmic phase of membrane. This is very important point, and never make any mistake. And another interlinking point with principles of inheritance and variation is regarding the human blood group antigens. Human blood group antigens A, human blood group antigen A, human blood group antigen B. Yes or no? Suppose this is A, B, and this is O blood group. This is A blood group, B blood group, AB blood group, O blood group. Yes or no? So you studied that. So what are these antigens? So these antigens are also sugar polymers, very clearly mentioned in principles of inheritance and variation. Human blood group antigens A and B and B, A and B. Don't write AB. There is no AB antigen. Many students are making mistake here. A antigen, B antigen, AB antigen they are writing. This is wrong. A and B antigens. You need to say like that only. There are only two antigens. So antigen A and antigen B present on the RBC plasma membrane. On the RBC plasma membrane means on which side they are present? They are present on exoplasmic phase because they are the sugars. They are present only on the exoplasmic phase. They are never present on cytoplasmic phase of the membrane. Concept you need to interlink. Okay. So this is what the important point is. Clear. And what is the nature of blood group antigens? Blood group antigen A and B, only for this I am saying. So they are sugar polymers, carbohydrates. But if you are talking about Rh antigen, that is not a sugar polymer, that is a protein. Okay, please remember this. Anyhow, in principles of redundancy and variation, we will study that. Next one, in human beings, the membrane of the erythrocyte, that is erythrocyte means what? RBC. RBC plasma membrane has approximately what percentage of protein and what percentage of lipid. It is a standard information. Scientists have already calculated, so you need to give the value. And please remember, this value is based on the weight percentage. From the membrane they have isolated proteins, from the membrane they have isolated the lipids, and they have measured the weight. For that weight, overall weight of the membrane and weight of the proteins. So based on the percentage by weight they have given this percentage. Not by number, don't get confused. So 52% by weight is the protein, 40% by weight is the lipids. For the human RBC, please remember, and again based on the location of the cell and function of the cell, and these percentages may be variable, but since they have already fixed this particular cell, this is the value, you need to compulsorily remember these values. Clear? Next one, type of plasma membrane, types of plasma membrane proteins based on ease of extraction. So we are talking about two types of membrane proteins. What are these two types of membrane proteins? One is called as peripheral or external or extrinsic proteins, and other type are integral, internal or intrinsic proteins. Two types are there. Please observe this information very clearly. What is the basis for this classification? Ease of extraction. What is the meaning of ease of extraction? How easily you can extract. Suppose this is the membrane and these are some proteins. I want to take these membrane proteins. I want to take these membrane proteins out of the membrane. I want to take these proteins out of the membrane. Then how easily I can take them out? So two options you will get. Easily you can extract, difficult to extract. Please remember, two options are there. One is easy to extract, other one is difficult to extract. But in NCERT directly they have given the names, but you need to understand the concept. Basis is ease of extraction. So if it is ease of extraction, you will get two options. Easy to extract, difficult to extract. Now tell me, which one are easy to extract? Those which are present on outer sides. Those which are present on outer sides are easy to extract. Understood or not? So that's why these are easy to extract. And those, please understand the concept here. I am directly telling the concept. Those proteins which are having interaction with hydrophobic tails. Those proteins which are having interaction with hydrophobic tails, they are intrinsic proteins. Please understand the logic. Some are thinking that present on outside, present on inside. So inside means up to where? So these are the problems. So simply understand the logic. If a protein is having interaction with hydrophobic tails, that means whether it is partially buried or completely buried, whatever it is, if some portion is having interaction with hydrophobic tails, it is difficult to extract. That's why they are called as internal integral proteins. Okay, internal or integral proteins or intrinsic proteins. But those proteins which are not interacting with hydrophobic tails, that means they are interacting only with hydrophilic heads. That means they are present on outer sides, so they can be easily extracted. So those proteins which are easy to extract, they are called as peripheral or external or extrinsic proteins. They are easy to extract. difficult to extract are called as internal or integral. So please understand the concept. Whether suppose they are giving one rough diagram, some animation diagram they make you, and they may ask you that, so which one is external, which one is internal. So observe carefully. The protein that is having interaction with hydrophobic tails is the integral or internal protein. So those which are not having interaction with hydrophobic tails, now they are external or peripheral proteins. This is the logic. Based on this logic only you need to decide it. Now what you are saying, some proteins like this is an integral protein because it is having interaction. Some integral, not all. This is not moving from one side to other side. Some integral proteins may extend from one side of the membrane to other side of the membrane. And such type of proteins are called as transmembrane proteins. Trans means across the membrane. So they are passing across the membrane. So these are called as transmembrane proteins. Now they may ask the question in reverse. Transmembrane proteins are integral proteins. True or false? It is true. All the transmembrane proteins are integral proteins. It is true. All integral proteins are transmembrane proteins. That is false. So observe the concept. Don't try to by heart all these things. Understand the concept so you can easily observe all these things. In this way, we are talking about different types of proteins. So again, depending on the nature of nature of the cell, location of the cell and its function. So different types of proteins are present in different cells. Clear? With that, these components are over. So lipids we have studied, proteins we have studied, carbohydrates we have studied, and then cholesterol also we have studied. So these are the 4 important points that we need to study under this cell membrane or plasma membrane. Now we are going with the structure of the plasma membrane. Before directly going with the structure, let us recollect the sentence from the previous lecture, that is the cell part 1. Under the prokaryotic cell, when we studied about plasma membrane, we have studied one sentence. The structure of plasma membrane for prokaryotes is similar to plasma membrane of eukaryotes. Why they are saying similar? Because the percentage wise for some of the proteins the structure may be slightly variable, but the basic phospholipid bilayer structure is present like that only. And Singren-Nicholson model, fluid mosaic model, is applicable to plasma membrane of prokaryotes and even for plasma membrane of eukaryotes. So that model is applicable everywhere. Clear? Based on the cell and the type, so some components may be variable. That's it. So that's why they said that the plasma membrane of prokaryote is similar to plasma membrane of eukaryote. That's what the point they said. Next one, plasma membrane is interacting with surrounding world. It is receiving some materials and releasing some materials. That's why cell is considered as open system. That point is applicable even for eukaryotic cell also. Yes or no? So in this way there are some important points that we need to understand. Next one, plasma membrane is selectively permeable, and here also we are saying that selectively permeable. So whether prokaryote, eukaryote, this basic structure and functions, they are almost same. Depending on cell and type, some small functions may be variable. Okay, so this is what. And now we are directly going with this fluid mosaic model. And who has proposed this? Singer and Nicholson. They gave the fluid mosaic model. In this fluid mosaic, please understand the two words, fluid and mosaic. If you understand these two words, you can easily understand the model. Fluid, the membrane is a fluid in nature. Actually, it is a semi-fluid in nature. Why we are focusing on this? Because even before these scientists, some other scientists have proposed different models of plasma membrane. But in those models, those scientists said that membrane is a rigid structure. If membrane is a rigid structure, you cannot explain some of the properties. So that's why, to explain those properties, these scientists have proposed that plasma membrane is a semi-fluid structure, and later on it is proved that it is true. So it is a semi-fluid structure. So that's why in that fluid mosaic model, the fluid is semi-fluid. Plasma membrane is a semi-fluid. What is the meaning of this semi-fluid nature? So here phospholipids are present as a bilayer. And now here what they are saying, because of this fluid nature, what is the fluidity of the membrane? Fluidity of the membrane is nothing but the ability of these molecules to move within the membrane. The ability of these molecules to move within the membrane. That means upper layer molecules will move within the upper layer. So lower layer molecules will move within the lower layer. So in this way, the ability of these molecules to move within the membrane is called as a fluidity. But actually it is a semi-fluid in nature. Clear? Or quasi-fluid. In NCERT they used a word called quasi-fluid nature. Clear? Next one is regarding the mosaic. What is the meaning of mosaic? Already you used to know about that tobacco mosaic virus. So what is that mosaic? Wherever it is, mosaic means it is patch-like. Mosaic is patch-like. What is present as a patches here? Proteins are present as a patches. Why it is important? Because in the previous models what they said is, they said that proteins are present as a continuous layer. In the previous models they said like this, but in this model what they said, they are not present continuously, they are present here and there like a mosaic pattern. So that's why fluidity is with respect to ability of these molecules to move within the membrane, and the mosaic they are saying because lipids are present continuously, but proteins are present here and there. So that's why it is a mosaic. So this type of model is called a fluid mosaic model. That's it. If you understand the meanings of these individual words, you can easily understand the model. And this model generally we are comparing with the icebergs that are floating within the sea. So here they may ask that what are these icebergs represent here? Here you are saying iceberg, but what are the icebergs here? So here the icebergs are nothing but the proteins. And here it is the sea made up of water. But here what is the sea? It is the sea of lipids. So that's why we say that they are the protein icebergs that are present within the sea of lipids. So in this way they are comparing this particular thing. Clear? So this is what about the fluid mosaic model. And anyhow you are talking, you may be studying about, anyhow in the regular lecture we studied more details. So horizontal movement and vertical movement, all the things we have studied. If these molecules are moving within the layer, within that layer, that is called horizontal movement. Yes sir, or lateral movement. Suppose they are moving from one layer to other layer, that is called as a vertical movement. And it is also called as a flip-flop. It is called as flip-flop. But regarding flip-flop, they have not mentioned in NCERT. Simply remember the name, that's it. So this is how they are present. This is what about the fluid mosaic model. So this is what the quasi-fluid nature. Because of the quasi-fluid nature, both proteins and lipids, they can move within the membrane. Within the membrane, they can move. Clear? This is what they are saying. And here, because of the fluid nature, we are able to explain various functions of the cell. Clear? Next one. So transport. So regarding the transport across the membrane, so huge information was not given here, very less information they give. Let us quickly go through this. Two types of transport we are talking here. So here plasma membrane is selectively permeable. Why you are saying selectively permeable? Why it is selectively permeable? Because it is not allowing all the molecules. Please remember, anyhow it will allow movement of water. Anyhow it will allow movement of water. Along with the movement of water, selectively it will allow only some of the solutes. All the types of solutes cannot pass through this. Selectively some type of solutes can pass through the plasma membrane. That's why it is selectively permeable. So please understand this. So semi-permeable means only it will allow the movement of water. Solutes cannot pass through semi-permeable membrane. But selectively permeable means water can pass through this and some solutes can pass through this. Freely permeable means all the molecules can easily pass through. That is called freely permeable. Impermeable means no molecule can pass through it. That's why generally you will get these are the four options: freely permeable, impermeable, semi-permeable, selectively permeable. These are the four options that you are having. You need to know the meanings of each and every option. Okay. Once again I am telling you, semi-permeable means it will allow the movement of only the water. Solutes cannot pass through this. But selectively permeable means water can pass through this and even some solutes can pass through this. That is called selectively permeable. So plasma membrane is selectively permeable. So you should not say that plasma membrane is semi-permeable. That is wrong. Plasma membrane is not semi-permeable, it is selectively permeable. Very important for examination. Okay. Now again, based on the movement of these things, now again different types of transports we are studying. One is called as passive transport, other one is called as active transport. In the passive transport, what happens? From higher concentration to lower concentration, molecules will be keep on moving. That's why it is called as movement along the concentration gradient. High to low is called as along the concentration gradient, and it is also called as a downhill process. Exactly opposite is for active transport. In the active transport, molecules will move from lower concentration to higher concentration. That's why it is called as against concentration gradient. It is called uphill process. Now you can easily understand. If it is a passive, anyhow they are coming from high to low, so energy is not required. So for passive transport, energy is not required. So ATP energy is not required. But for active transport, since they are moving from less concentration to higher concentration, so energy is required. So these are some basic important points that we need to understand. And within the passive transport, again we are talking about different types. one is the simple diffusion. In the simple diffusion, membrane proteins are not required. Simply across the membrane, based on the concentration gradient, the molecules will move from higher to lower. So here membrane protein is not required, energy is not required. Facilitated diffusion, anyhow energy is not required, but a membrane protein is required. They cannot move on their own. So what type of molecules will do this? Simple diffusion, hydrophobic molecules mainly. Okay, hydrophobic molecules, they are moving through simple diffusion, because through the membrane hydrophobic molecules can easily pass through, hydrophilic molecules cannot easily pass through. So who will pass through this facilitated diffusion? Hydrophilic molecules. Hydrophilic neutral molecules, neutral molecules can go through simple diffusion, but charged molecules, charged ions, they have to pass through by facilitated diffusion if they are moving from higher concentration to lower concentration. Next one is regarding osmosis. Osmosis is also a type of facilitated diffusion. Please remember, osmosis is also a type of facilitated diffusion, because here also membrane protein is required, but this term is exclusively used for movement of water. For the water movement, we are using the term called osmosis. So for the movement of water across the membrane, a membrane protein called aquaporin is required. So that's why osmosis is like a facilitated diffusion, but exclusively used for movement of the water. In the osmosis also, endosmosis, exosmosis, various concepts, examples, applications, so many things, so many important things for examination we have discussed in the regular lecture. Okay, please go through that point once again. Clear? This is what. The next one is regarding the active transport. Anyhow, it requires energy. Movement of molecules is from lower concentration to higher concentration. That's why it is called as against the concentration gradient. And here again it is divided into two types, primary active and secondary active transport. In the primary active transport, ATP energy is directly used. One important example given in NCERT is Na plus K plus pump. Okay, if 3 Na plus are pumped out of the cell, 2 K plus will be pumped into the cell for the same thing. Okay, so in this way, so in the secondary active transport, what happens is energy is used indirectly. Okay, so the gradient created by one may be used for other one. So in this way, ultimately energy is required, but ATP may not be directly used in this particular case. So this is how about the transport. And then now we will simply go through this particular one. Anyhow, this was deleted in the new NCERT, but since this table is related with this particular content, so better go through this. Requires special membrane protein. Does simple diffusion require any special membrane protein? No. Without any membrane protein, directly they can cross through. So no. But facilitated diffusion and active transport, yes, they need membrane protein. Next one, selective. Here there is no membrane protein, so that's why it is not selective. But these two are involving membrane proteins. So membrane proteins are highly selective. They can only transport specific type of molecules or ions. That's why they are highly selective. So that's why facilitated and active transport, they are selective process. Next one, transport saturates. When a transport saturates, if membrane protein is involving, the transport can be saturated. Suppose there is a one protein can carry only suppose 4 molecules. Suppose, let us take example. Suppose more than 4 are there, then what happens? It can take only 4 at a time. At a time, it can take 4. That means 4 is the saturation level. That means it got saturated. But in simple diffusion, there is no membrane protein. So there is no concept of saturation. So concept of saturation applicable to facilitated transport and active transport only. Next one, uphill. Simple diffusion is downhill. Facilitated diffusion is also downhill. So which one is uphill? Only active transport is uphill process. Next one, requires ATP energy. Simple diffusion, facilitated transport, they are part of passive transport. So they don't need ATP energy. But active transport, yes, it needs energy. Clear? This is what. So with that, the cell membrane and its functions, all the things we have completed. Now let us quickly go through these cell organelles. And in the case of eukaryotes, different types of cell organelles are present. And here we are classifying them based on the membranes that they are present. Some organelles have double membrane. 2 membranes are present. 2 membranes means 4 layers of phospholipids. Please remember, what are those? Nucleus. Nucleus is double membrane bound organelle. Mitochondria also, chloroplast also. These are the 3 double membrane bound organelles. Next one, single membrane bound organelles. What are the examples under this? Endoplasmic reticulum, Golgi complex, lysosomes, vacuoles, microbodies. Under the microbodies, what you can say? Peroxisomes, glyoxysomes, spirosomes, all these things will come. But anyhow, spirosomes, glyoxysomes names were not mentioned in NCERT. At least peroxisomes names you please remember. Okay, remember. Very good. Next one, non-membrane bound. There is no membrane around them. What are those organelles? Centrosome and ribosomes. So these are the cell organelles. Okay, clear. Next one, Inside the nucleus, generally we are talking about the nucleolus. Under the organelle, we are not counting it, but inside the nucleus, nucleolus is there, but that is also not having any membrane around it. Okay, so these are some important points that we need to remember. Next one is regarding endomembrane system. What is the meaning of this? Endo means what? Inside. Inside what? Inside the cell. Inside the cell, membrane system is there. That means there are some membrane bound organelles that are functionally interrelated with each other. Inside the cell, some membrane bound organelles are functionally interrelated. So all those things combined, they are called as endomembrane system. So what are present in the endomembrane system? Endoplasmic reticulum, Golgi complex, lysosomes and vacuoles. These are the four things that are present in the endomembrane system. And please remember, some students, they are thinking that plasma membrane is also part of endomembrane system. That is wrong. Plasma membrane is not part of endomembrane system. Inside the cell means inside the plasma membrane. So why you will consider the plasma membrane? You should not count the plasma membrane. Please remember this. Plasma membrane will come under a separate one, that is called a secretory pathway. That is a different thing. That is not there in your syllabus. Okay. So endomembrane system is having only four organelles: endoplasmic reticulum, Golgi complex, lysosomes and vacuoles. That's it. Okay. In NCERT they said that mitochondria, chloroplast, peroxisomes, all these are functionally not related with this. Functionally they are not related with this. That's why these mitochondria, chloroplast, peroxisomes, all these are not part of endomembrane system. That's why only these four are present in endomembrane system. Very good. Four organelles are there. Up to that it is good. But now you need to read the question very carefully. Endomembrane system of plant cell, if they ask, all the four are there. So you can write all the four names. Suppose they are talking about endomembrane system of animal cell. In animal cell, endoplasmic reticulum, GC, lysosomes, these three are present. But vacuoles are not present in animal cells. So if they are specifically asking about animal cell, then you need to write only these three. Understood or not? So this is what one important point that you need to notice here. Next we will talk with individual organelles, and first we will focus on endoplasmic reticulum. Endo, inside, plasmic, cytoplasm, inside the cytoplasm, reticulum, network like organelles are present. That is the endoplasmic reticulum. Simply meaning it is. So it is the network or reticulum of tiny means a small tubular network like structures that are distributed in the scattered means they are distributed in the cytoplasm. So this is how they are present, and it is called as endoplasmic reticulum. Again, this endoplasmic reticulum is classified into different types. And before going with this, first we will understand what are the names of what are these words and their meanings. So luminal side, extraluminal side. What is the meaning of this? Suppose this is the endoplasmic reticulum. So this is the membrane. This membrane is enclosing some space. The space enclosed by this membrane is called as a lumen. So that means luminal means what? Inner side of the endoplasmic reticulum. So extraluminal means what? Outside the endoplasmic. Outside endoplasmic reticulum means what? Cytoplasm side. So that's why extraluminal side means cytoplasmic side. Luminal side means towards inner side of the endoplasmic reticulum, towards the lumen side. Understood or not? These are some words. Why we are talking about these words? Because immediately we are going with types of endoplasmic reticulum. Rough endoplasmic reticulum is having the ribosomes. On which side they are present? Towards the lumen side they are present, or towards the extraluminal side they are present. This is very important thing. So please remember this. In the case of rough endoplasmic reticulum, ribosomes are present only on outer side. Outer side means extraluminal side. Extraluminal side is also called as cytoplasmic side. Very important point. Okay. So please remember this. And since ribosomes are present, what is the function of ribosomes? Protein synthesis. So what is the function of rough endoplasmic reticulum? Protein synthesis. This is what the point is. But the next important point is, observe carefully. Rough endoplasmic reticulum is frequently observed in the cells that are actively involved in protein synthesis and secretion. Cells that are actively involved in protein synthesis and secretion. This word is important. Sorry, this is not just a random word. You need to give more emphasis on this word, secretion. Protein is secreted from the cell. Now this needs some explanation. The length of this video may be increased, so don't worry. Let us try to understand these points. Why we are focusing on the word secretion? Now observe carefully. Already in the previous lecture I told you what are the locations of ribosomes in eukaryotic cell. Some ribosomes are present freely in cytoplasm. Suppose this is the nucleus. Some ribosomes are attached on outer membrane of nucleus. And suppose this is endoplasmic reticulum. Suppose endoplasmic reticulum. Some ribosomes are attached on endoplasmic reticulum. Suppose this is the mitochondrial matrix. Mitochondrial matrix. Inside this also ribosomes are present. Suppose it is a plant cell, chloroplast will be there, and inside the chloroplast stroma also ribosomes are present. These are the 5 locations we have studied. Now observe carefully. Now what they are saying? Cell is actively involving in protein synthesis and secretion. So protein is secreted from the cell. Protein is secreted from the cell. That means a protein is released outside the cell. If a protein is released outside the cell, now the question is, which ribosome will synthesize this? A protein that is released outside the cell is synthesized by which ribosome? Free ribosome, ATS type. I am giving you options. 5 options are there. Free ribosomes, ATS type, and ribosomes attached on outer membrane of nucleus, that is also ATS type. Ribosomes on endoplasmic reticulum, this is also ATS type, and ribosomes of mitochondrial matrix, 70S type, and ribosomes of chloroplast stroma, 70S type. These are the 5 options. Now tell me, what is the correct one? So which ribosome is involving in synthesis and secretion of protein from the cell? This is very important point. It is the ribosome that is attached to endoplasmic reticulum. If a protein is released outside the cell, means it should be synthesized by ribosome attached on endoplasmic reticulum only. It is fixed. Please remember this. So now you need to understand that ribosome attached to endoplasmic reticulum will synthesize proteins for whom? This is also another very important question. You need to understand this concept only. Remaining points anyhow you can easily understand this. So this ribosome present on ER will synthesize, first one, proteins for ER. Proteins for ER are synthesized by the same ribosomes. Next one, proteins for Golgi complex, they are also synthesized by ribosomes on endoplasmic reticulum. Proteins for lysosomes, they are also synthesized by ribosomes on ER. Proteins for vacuoles, they are also synthesized by ribosomes on ER. You can easily understand this. These 4 are part of endomembrane system, right? These are part of endomembrane system. So proteins required for endomembrane system are synthesized by ribosomes on endoplasmic reticulum. And not only this, proteins required for plasma membrane. Some proteins are present in plasma membrane also. They are also synthesized by ribosomes on endoplasmic reticulum. And if cell wall is there, if cell wall is present on outside this, and in that case, cell wall is also having some proteins, and who will synthesize those proteins? They are also synthesized by these ribosomes only. And finally, it is the secreted proteins. Proteins that are secreted outside the cell, these are also produced by ribosomes and endoplasmic reticulum. This is a very important concept. So please remember this. You should never make any mistake. I will give you one simple example. Insulin hormone. In the chemical coordination and integration chapter, we have studied about insulin hormone. Insulin hormone is released outside the cell. And what is the nature of this insulin? It is a protein hormone. Yes or no? In biotechnology applications also we studied this. So insulin is a protein hormone. So this hormone is released outside the cell. That means it is a protein that is released outside the cell. So insulin is produced by which ribosome? Ribosome present on endoplasmic reticulum. Glucagon, that is also a protein hormone, that is also produced by ribosome on endoplasmic reticulum. So in this way, this concept may be linked with a particular example. This is simply a basic concept. So you should not be confused. Link up this. So you can easily get the answer. So in this way, this is what they are saying that. So this endoplasmic reticulum is more actively present in the cells that are involving in protein synthesis and secretion. The word secretion is very important for examination. Okay, there is a huge concept behind this single word. Okay, this is what, and it is extensive and continuous with outer membrane of the nucleus. So it is continuous with outer membrane of the nucleus. Clear? Next one is regarding smooth endoplasmic reticulum. Generally it is present away from the nucleus, and here ribosomes are not present. So since ribosomes are not there, it is not involving in protein synthesis. Instead of this, it is involving in synthesis of glycogen. The word glycogen was given in the summary. So please remember, smooth endoplasmic reticulum is involving in glycogen synthesis. In the summary they have mentioned this word. So please understand this. Along with this, it is involving in synthesis of some steroid hormones in the case of animal cells. Okay, and next one, it is majorly involving in the synthesis of lipids. So these are the various functions of this smooth endoplasmic reticulum. Next one, sarcoplasmic reticulum. In the locomotion and movement chapter, we studied about this. So sarcoplasmic reticulum is nothing but endoplasmic reticulum that is present in the muscle cell. In the muscle cell. And what is the function there? It is storing the calcium ions that are required for the muscle contraction. So sarco means muscle. So it is the endoplasmic reticulum present within the muscle cell. Please understand this. Next one is regarding the Golgi apparatus or the Golgi complex or the Golgi bodies. Any of these words you can use it. And these were discovered by Camillo Golgi. So based on his name, we are calling them as Golgi bodies. So generally in the case of plant cells, they are distributed because central vacuole is present in plant cell. So that's why these Golgi bodies are distributed there. And there we are using a term called as dictyosomes, but that name was not mentioned in NCERT. Next one. These are densely strained reticular structures. These are also reticular network-like structures. They may be continuously connected with each other or they may not be connected, but functionally they are distinct. Okay, so they are having at different locations, different functions will occur, but all these are working together, functioning together. So in this way they are present. These are flat disc-shaped structures and they are having different sizes and they are present stacked parallel to each other. They are present one above the other, parallel to each other. Sometimes they are interconnected, sometimes they are not interconnected, but functionally they are having specific functions, distinct functions are present. And here also some phases we are talking. Cis phase we are saying on one side, trans phase we are talking on the other side. Okay, cis phase will receive. Cis is also called as a convex or forming phase, whereas trans phase is called as maturing or concave phase. Cis phase is receiving materials from where? From the endoplasmic reticulum. I told you, no, from endoplasmic reticulum, it will go to the material will go to the Golgi complex. From the Golgi complex, diversion. It may go to lysosomes or it may go to vacuole. So in this way, the cis phase is receiving materials from the endoplasmic reticulum in the form of vesicles. From ER, some vesicles will come out with some materials. They will come and fuse with cis phase. And slowly they are passing through this medial Golgi, cis phase. And this is the trans phase. And the middle part is called a median Golgi. It is slowly passing through this. When it is passing through this, it is undergoing some chemical modifications, chemical changes. And finally, it is reaching the trans phase. Once it reaches the trans phase, by the time it will be that whatever is the modification it is undergoing, that will be completed so that it can be easily released from this particular trans phase. If it is going to lysosome, it will go. If it is for vacuole, it will go there. If it is for plasma membrane, anyhow it is not part of endomembrane system, still it will go. I already told you, know, it can go to plasma membrane and there it can go to different other locations. So this is what about this particular phases and all. Clear? Next one. So in this way, the movement all these things we have studied. And what are the important modifications that we are talking in the Golgi complex? Glycoproteins, glycolipids, all these things. Now you need to understand one point. Proteins from rough ER, lipids from smooth ER, they are coming to GC. So this protein and lipids, they are coming from endoplasmic reticulum. Now what is happening in the GC? Glyco is added. That means within this Golgi complex, glycosylation is taking place. What is the meaning of glycosylation? Glyco means carbohydrate portion, right? So that carbohydrate portion will be added. So carbohydrate added to protein, it is called a glycoprotein. Carbohydrate added to lipid, so glycolipid will be formed. So that's why this Golgi complex is the major site for glycosylation, means attachment of carbohydrates. That is what the important point that they are talking. And this Golgi complex is principally involving in packaging of materials and distributing to their targets. So in this way, at the trans phase, once the material is matured, what happens? Now it will be packaged in the form of a vesicle, and wherever it has to go, there it will distribute it. So this is the overall information that they are talking about this particular Golgi complex. Next we will go with the lysosomes. So lysosomes from the Golgi complex, just now we said, no, from the Golgi complex, this vesicle, if it is having material for lysosomes, it will go and form lysosomes. So in this way, newly formed lysosomes are called as a primary lysosomes, and they are having some enzymes. So these enzymes have the ability to digest almost all major biomolecules like carbohydrates, lipids, proteins, nucleic acids. So that's why what are the enzymes that are present? Protein digesting enzymes are proteases. Lipid digesting enzymes are lipases or lipases, anyhow you can pronounce. And nucleic acid digesting enzymes are nucleases. And then finally what is left? Carbohydrates. Carbohydrates, they will break down the carbohydrates. So in this way it is having various hydrolytic enzymes. So these enzymes will break particular bond with the use of water molecule. That's why they are called hydrolases, hydrolytic enzymes. And these enzymes are active at acidic pH of approximately 4.5 to 5 pH, they are active. So this is what some basic information that they are talking here. So newly formed are called as primary lysosomes, and whenever a food is engulfed, it will form the phagosome, and the phagosome and the primary lysosome will form, and then now it will start digesting it. So in this way, once these two are attached, it is called phagolysosome, it is called secondary lysosome, and the enzymes will start digesting the food, and all the nutrients will be taken out, and the waste will be kept, and that lysosome having the waste is called residual body or tertiary lysosome, and it will go and fuse with the cell and exocytosis will occur. So this is how these lysosomes are very important. Next one is regarding the vacuoles. So these vacuoles are some spaces that are present inside the cell, and these vacuoles are of different types, but our main focus is on plant sap vacuole. So initially we are talking about the plant sap vacuole. So what is the sap vacuole of the plant? Mainly it consists of water, sap, some excretory products, means they are not useful for the cell, so they will be stored in that, and some other materials that are not useful for the cell. And please remember, and these vacuoles in some particular types of plants, so they are storing even some secondary metabolites also. They are not useful for the plant, but they may be useful to the human. Okay, so they can also be extracted from this. Next one. So this vacuole is having a single membrane, and the membrane of the vacuole is called as tonoplast. And the content present inside the vacuole is called as tonoplasm. Tonoplasm is the content inside the vacuole. Tonoplast is the membrane of vacuole. So please remember this. In the case of plant cells, this vacuole can occupy up to 90% of the volume. So that's why the largest cell organelle of plant cell, if they ask, it will be the vacuole. In general they are asking, then we need to talk about nucleus, because in the case of animal cells, vacuole is not there. So in that case, the largest cell organelle will be nucleus. So please remember this one. Okay, next one. So here in the case of vacuole, what happens is, suppose this is the surrounding cytoplasm. Now here what happens on this tonoplast membrane, the membrane is called tonoplast, right? On this membrane there are some pumps, and what these pumps will do, by spending energy, please remember the term pump we are using for active transport. The term pump we are using in active transport, and the term channel we are using in the passive transport, and mainly for that facilitated diffusion. For the facilitated diffusion, passive transport, the membrane protein is called as a channel, and the membrane protein that is involving in active transport is called as a pump. So these are technical words. I am using a term called as a pump. Pump means it is involving in active transport, using the energy from lower concentration to higher concentration. Molecules and ions will pass, yes or no? So that's why there are some pumps that are present in the tonoplast, and using some energy, the molecules and ions from the cytoplasm will be taken into the tonoplasm. So that's why inside this vacuole their concentration will be increased, so that they are accumulated. So this is how it is maintaining the higher concentration when compared with cytoplasm. This is what they are talking here. This is what the information that I have given here. Next one is regarding contractile vacuole. So generally we are observing in some lower animals like amoeba, which are living in the fresh water. Please remember, living in the fresh water. So in the case of fresh water, the problem is the concentration of the cell is more when compared with the surrounding fresh water. Concentration of fresh water is very less. So because of the osmosis, what happens? Water from the outside will enter into the cell. So that's why water from the outside will enter into the amoeba here. So now what happens? Its sizes keep on increasing because continuously water is coming. So its sizes keep on increasing. If the excess water is not released, now it may burst and die. So that's why what happens? There is a contractile vacuole. Excess water will go into the contractile vacuole. Not only excess water, some waste materials are also going into the contractile vacuole. And once the contractile vacuole size is increasing, and it will fuse with this plasma membrane, and excess water and waste will be released outside the cell. So again, the cell will get its original shape or original size. Okay, anyhow it is not having a fixed shape, but the thing is the original cell concentration will be maintained. So in this way, contractile vacuole is involving in osmoregulation and also excretion. Very, very important point. Please remember this one. And next one is regarding the food vacuoles. So in many cells like protease, what they do is they will engulf some food material. Once they are engulfing food material, they are forming the vacuole that is called as a food vacuole, or you can also call it as a phagosome, food vacuole. Now anyhow, this food vacuole again combines with that lysosomes and then digestion will occur, all these things. But our focus is about only food vacuole. So food is taken in the form of a vacuole. So that's why it is called as a food vacuole. Clear? And next one is regarding the mitochondria they are talking. In the NCRT, they have given a sentence. Unless it is specifically stained, we cannot clearly see this under microscope. Please observe this. Unless specifically stained, you cannot see. That means its size is very, very small. And second point is, if you want to see mitochondria, you need to specifically stain the mitochondria. So for the specific staining of mitochondria, that means, so here if you are using the Janus green dye, it is a dye. So if you are using this Janus green dye, what happens? It will specifically stain mitochondria only. Other organelles cannot be stained. So when you are observing under microscope, whatever is the stained organelle is there, that itself is the mitochondria. So you can easily locate this particular mitochondria under the microscope if you are using the specific dyes. This is what the point they are saying. And this example, though not mentioned in the regular NCRT, it was given in NCRT QR code content. So every student should compulsorily remember this particular name. Okay. So this is the Janus green is the stain that is used for mitochondrial specific staining. So if you are using this, then you can easily observe under microscope. Next one, number of mitochondria. There is no fixed number. So what is the function of the mitochondria? Mitochondria will provide energy, ATP energy. If a cell requires more ATP, in that cell, more mitochondria will be there. So that's why based on the physiological activity of the cell, number of mitochondria will be variable. So even in our body also, don't think that there is a fixed number in all the cells. No. Again, in different cells, different physiological activities will occur. The cell that is more actively present, that will have more mitochondria. The cell that is having less activity will have less number of mitochondria. Next one regarding the shape, there is no fixed shape. It is having various shapes, but the more generally observed shape is sausage shape. You can also call it as cylindrical shape. Okay, this is how it is present. Sausages are cylindrical shaped, and they have given some size also. Observe it carefully. And it is a double membrane bound organelle. So here two membranes are there. Two membrane means overall how many phospholipid layers? In this two, in this two. So total 4 phospholipid layers are present. Yes or no? And between these two there is a space. So anyhow, one by one we will go through all these things. So this is the outer membrane. This outer membrane forms the continuous limiting boundary for the organelle. And between outer and inner membrane there is a space that is called as perimitochondrial space, or in general you can call it as intermembranous space also, you can say it. And that is also a aqueous compartment. What is the meaning of aqueous compartment? Mainly it is filled with water. And not only this, some other components are also present. And inner membrane is not smooth, and actually it is having some finger-like projections, and these finger-like projections are called as cristae. Wherever foldings are there, already we studied that. So this will increase surface area. What is the advantage of increasing surface area? So if surface area is increased, then what happens? So more and more F0, F1 particles can be accommodated, and these are actually involving in ATP synthesis. If more surface area, more ATP can be synthesized. That's why they are having these foldings. Yes or no? Next one. So here what we are talking. So this inner membrane is enclosing one aqueous compartment, but inside this it is having some semi-solid dense material that is present. It is called as a matrix. And in this matrix, so many components are there. Some of the important things that we need to mention are DNA molecules are there. In the NCRT, they have mentioned that single circular DNA molecule they said, but please remember, not just one copy. Exactly same, many copies can be present. Please understand this. In the NCRT, there is a problem here. Here they said that for the mitochondrial matrix, they said single circular DNA molecule. But for chloroplastostroma, they said many circular DNA molecules, they said. So please remember, for both of them, for mitochondria and chloroplast, for both of them, please remember many same copies of circular double-stranded DNAs are present. Okay, so please understand this particular point. And DNA is there. From the DNA, upon transcription, RNA will be produced. So RNA is also present, and from this RNA, and here protein will be produced because 70S ribosomes are present. So all these are present. So in this way, many things are happening in this particular one. And regarding this mitochondria, so what is the main function of this one? It is involving in aerobic respiration. So under the plant physiology, there is a separate chapter called as respiration. There you will study more details with complete mechanism. So since it is involving in aerobic respiration and involving in ATP synthesis, mitochondria are called as powerhouses of the cell. Clear? So this is what the information regarding this particular one. Oxysomes are the ones through which H plus ions are passing through so that ATP energy can be produced. Anyhow, more details you will study under respiration chapter. And mitochondria will divide by fission. Please remember, this is one property that is similar to prokaryotes like bacteria. Yes or no? So this is one important point. And it is called as semi-autonomous organelle. Semi-autonomous. Why it is called semi-autonomous? What is the meaning of autonomous? Autonomous means self-existing. Self-existing. But the question here is, can mitochondrium, already I told you in the previous lecture, can individual organelle have independent existence? No, it cannot exist independently. So that's why mitochondria are not autonomous, but they are semi-autonomous, of autonomous. So of self-existing. Why you are saying of self-existing? Because it is having its own DNA, its own ribosome, so it can synthesize its own proteins. So to some extent it is able to synthesize its own things, but can it live independently? No, it cannot live independently. Please don't get confused here. So that's why it is called as semi-autonomous organelle. Clear? Next one we are talking about the plastids. Plastids are found in plant cells and even in some euglenoids also it is present. Clear? And what are these plastids and what are the types? So these plastids, based on the type of pigments that are present, we are classifying into three types. First one we are talking is the leucoplasts. These are colorless plastids. They don't have any pigments. They don't have any colored pigments. So that's why leucoplasts. And chromoplasts and chloroplasts. These are having some pigments. So we will go one by one. Leucoplasts. So leucoplasts are the colorless plastids. They don't have any pigments. So again these leucoplasts are divided into three types based on the type of material that is stored. Generally leucoplasts are involving in storage of some nutrients. So what type of material that is stored? Based on this again three types are there. Amyloplasts. Amyloplasts. What are amyloplasts? So amylo means it is carbohydrate. So they are storing the carbohydrates. So that's why these are called as amyloplasts, and like starch will be stored. One standard example is potato. Next one is LIO+. What they are storing? They are storing oils and fats. Next one is Eluroplast. They are storing the proteins. So in this way, based on the type of material that is stored, these leucoplasts are of different types. Now what type of question they may ask? What is the color of leucoplast? Colorless. You can write directly. But the thing is, sometimes they may ask that, what is the color of amyloplast? What is the color of elioplast? What is the color of eliroplast? Like that they may ask. Still you need to write colorless only. Many students are getting confused there. So you need to write colorless only. Now come to the next one, that is regarding the chromoplast. Chroma means what? Color. So these are having some colored pigments. But the pigments other than the green color, because green will come under the chloroplast. Clear? Now here what are the different types of pigments that are present? Carotenes are present, xanthophylls are present, likewise different types of carotenoid pigments are present. Clear? So in this way different types of pigments are present, and because of these pigments, so they are giving color like yellow color, orange color and red color. So these are the colors that we are generally observing because of the chromoplast. Finally come to the chloroplast. Chloroplast is having chlorophyll and also it is also having some carotenoid pigments. So carotenoid pigments are present in both chromoplast and chloroplast. Please remember this. Carotenoid pigments are present in both chloroplast and chromo and chloroplast. Both of them are having this. But chlorophyll is present only in chloroplast. This is one important point. Next important point is, what is the nature of these pigments? These chlorophyll, carotenoid pigments, all these things, xanthophylls, carotenes, what is the nature of these pigments? These are lipid soluble pigments. Please remember this. So plastids are having, if they are having the pigments, plastids will have what type of pigments? Lipid soluble pigments they are having. Lipid soluble pigments they are having. Please remember this. Why I am focusing on this one? Because sometimes, even though not mentioned in NCERT, even in the vacuole also some pigments are present. If you are observing this, yellow, orange, red, and in this case it is the green color, right? And these are due to lipid soluble pigments present in the plastids. But if you are observing some pink color, violet color, for some of the fruits or flowers, you are observing these colors, and those colors are due to some pigments present in the vacuole. Please remember this. Pink color, violet color, blue color. Yes or no? So these colors are mainly due to some pigments that are present in the vacuole. So vacuole is mainly having water. So that's why they are water soluble pigments. The pigments that are present in the vacuole are water soluble pigments, but the pigments in this are fat soluble pigments. Small difference is there. You need to understand these differences. Clear? This is what. So different colors are possible because of different types of pigments that are present in the plastids. Clear? Next one, regarding the chloroplast, we are going with more details and structure. So majority of the chloroplasts we are observing in the green plants, and these are mainly we are observing in the leaves, mesophyll layer of the leaves. So this is what the basic structure, and before going with this, we are focusing on various shapes. In NCRT, they have mentioned about, so disc-like they have mentioned, ovoid, disc-like, round. Likewise, they have mentioned majority of these shapes are related to higher plants. Clear? Next one. Another example they have mentioned in NCRT is ribbon-like. Please remember this. These words were used in NCRT, so you need to remember this. In NCRT, they said ribbon-like chloroplast. Ribbon-like is also called as spiral shaped. And in which organism we are observing this? In Spirogyra, we are observing this. This is another example that you need to remember. Next one. Regarding the number of chloroplasts per cell, they said that only one chloroplast is present in one cell. One cell is having only one chloroplast, and further they gave the example of chlamydomonas. And for this chlamydomonas, there is a single chloroplast. And what is the shape of this single chloroplast? It is the cup shaped. For the chlamydomonas, in single cell only single chloroplast is there. That name was mentioned in NCRT. But what is the shape? They have not mentioned. But at least name, number they have mentioned. Simply add one more point. What is the shape it is? It is the cup shape. At least you please remember this. So now we will come back to all these things. Cup shaped chloroplast is present in chlamydomonas. This is one important point that you need to remember. And next one, spiral or ribbon-like is present in Spirogyra. And next one, discoid or oval-like, it is present in the higher plant. At least these are some basic important, and remaining things are just understanding purpose only. Clear? And regarding the number, only single chloroplast is present in one cell. For that example is chlamydomonas, which is a green alga. Okay. Next one, if you are talking about the, so higher plants, if you are talking, so they are having 20 to 40. Okay. So this is what the information is. Next one is regarding the chloroplast structure. If you are observing, this is also a double membrane bond, outer membrane, inner membrane. Okay. So now between these two also there is a space. This is called as periplastidal space or perichloroplastidal space, or in simple intermembranous space. Any name you can use it. Next one. So inner membrane is enclosing a space that is called as a stroma, and in this stroma what happens? Dark reaction will occur. That means carbohydrate synthesis will occur. And in this stroma what is present? DNA is present. Circular double-stranded DNA molecules, many but same copies are present. From the DNA, RNA is produced. So RNA is also there. From the RNA, protein is produced because 70S ribosomes are present. So all these things are present within the stroma. Now along with all these things, now there are some sac-like structures, they are called as thylakoids are present. And how these thylakoids are present? Thylakoids are present in two forms. Now better I need to write this because many students are making mistake here. Thylakoids are present as grana or intergranal thylakoids. Intergranal thylakoids. Many students are making mistake here, so that's why I am giving more explanation. Many students are thinking that grana, intergranal thylakoid, these are alternative names they are thinking. They are thinking that grana are also called as intergranal thylakoids, but this is wrong concept. Grana are not called as intergranal thylakoid. Meaning is different. Inter means between what? Grana. Between grana. Here they are grana. Here between grana. How they will be same? They are not same. So please observe the diagram. Suppose this is the inner membrane, this is the outer membrane of the chloroplast. What they are saying is these are the flat sac-like structures that is called as a thylakoid. How thylakoids are present? Thylakoids are present as grana. This is one. Or thylakoids are present as intergranal thylakoid. This is what the meaning is. They are two separate things. Thylakoids are present as grana, or thylakoids are present as intergranal thylakoids. This is what the meaning is. Now we will go with the first option. What is this? Thylakoids are present as grana. What are the grana? Grana are the stack, means like pile of coins. Now they are present one above the other. So this is one complete pile and this is called as granum. Singular is granum. Suppose there is one more like this. This is also granum. If you are talking about both of them, plural, under the plural what you will say? Grana you will say. So in this way thylakoids are present as grana. This is the first one. Or they are present as intergranal thylakoid. Inter means between the grana also thylakoids are present. What is this? This is one granum, this is one granum. Between the grana also thylakoids are present. Understood or not? So these are grana. So thylakoids are present as grana or thylakoids are present as intergranal thylakoids. So this is what the point is. So don't make mistake here. So this is what they are saying. So in this way thylakoids are present here. Clear? And after this we are talking about the stroma lamella. So there are some extensive tubular membranous tubular structures that are connecting different grana. They are called as stroma lamella. But please remember in this chapter they said intergranal thylakoid separately and stroma lamella separately, but technically both of them are same only. In this chapter, once again I am repeating, in this chapter, that is cell the unit of life, they are talking intergranal thylakoid separately and stroma lamella separately, but technically both of them are same only. Okay, so these are the connections between the different grana. Clear? Next one, what is the lumen of thylakoid? So inside this each thylakoid there is a space. That space is called as lumen of thylakoid. And this thylakoids are involving in photosynthesis because they are having the pigments. So in this way light energy will be trapped and it will be used for synthesis of chemical energy. So this is what they are saying. So within the stroma there is enzymes for the synthesis of carbohydrates because dark reaction is taking place there. And then DNA molecule are present, RNA molecules are present, ribosomes. Which ribosome? 70S ribosome. 70S are called as smaller ribosomes, 80S are called larger ribosomes. So which ribosomes are present in chloroplast? 70S ribosomes are present. And mitochondria are also called as semi-autonomous organelles because they have their own DNA and they have their own ribosomes for protein synthesis. That's why they are called as semi-autonomous organelles. And here you need to understand before going with this. First I will give some relationship between bacteria, mitochondria and chloroplast. Then we will go with endosymbiotic theory. Okay. So all these three if you are observing, in the case of bacteria here type of ribosomes. What are the type of ribosomes in bacteria? 70S. Mitochondria is also having 70S. Chloroplast is also having 70S type of ribosomes. Regarding the DNA if you are talking, here it is circular double stranded DNA. Circular double stranded DNA. Here also the same, here also the same. Yes or no? In all these things, circular double stranded DNA is present. And regarding the histones, DNA is present. But is the DNA associated with histones? No. Histones are not present here, histones are not present here, histones are not present here also. Understood or not? Next one is by the division. How it will divide? Bacteria will divide by fission. And here mitochondria is also dividing by fission and chloroplast is also dividing by fission. So please understand this. So for majority of these properties, mitochondria and chloroplast, they are resembling the bacteria. Mitochondria and chloroplast are resembling the bacteria. So in this way, now we are talking about a hypothesis that is called as endosymbiotic theory. And what this endosymbiotic theory says, in the beginning days of evolution, what they are saying, so there is a free living organism. So now it has a free living bacterium. So it has engulfed some chemosynthetic bacteria. So that now that chemosynthetic bacteria slowly became mitochondria. This is what a hypothesis that they are saying. Clear? So in this way that's why they are saying that there is a relationship between mitochondria and the bacteria. And in this way it has engulfed a photosynthetic bacteria, so that now it is resembling chloroplast. So chloroplast is resembling with the bacteria. So in this way, for some of the properties, they are comparing this mitochondria and chloroplast with the bacteria, and to give this comparison, so they have given this particular theory that is called as endosymbiotic theory. So these bacteria, they become part of the eukaryotic cell, initial eukaryotic cell, and then now there is a symbiosis. So this mitochondria and chloroplast, they are providing something to the eukaryotic cell. So in turn eukaryotic cell will provide something to these organelles. In this way there is a symbiosis between these two. So present inside the eukaryotic cell and then maintaining symbiosis. So that's why this theory is called as endosymbiotic theory but not there in NCERT, but if you can understand, it will be better. That's it. That's why I don't want to go into more details. Next one is regarding the ribosomes. So ribosomes are granular structures, and these are also called as Pallade particles because they were observed by George Pallade. Next one, what actually these ribosomes are? Already in the previous lecture I told you. So these are RNA molecules with some proteins, simply RNA and proteins. There is no membrane around this. That's why these are non-membrane bound organelles, not surrounded by any membrane. And here, whether it is the two types of ribosomes we are studying, 70S ribosome, 80S ribosome. But please remember, a higher level concept, there is one more concept, 55S ribosomes. I told you they are present in mitochondria of mammals, but that concept is not needed for you, so leave it. We are talking only about 70S and 80S. 70S ribosomes are called smaller ribosomes, 80S are called as larger ribosomes. 70S are called organellar ribosomes and 80S are called as cytoplasmic ribosomes. 70S are called as prokaryotic ribosomes and 80S are called as eukaryotic ribosomes. These are some basic names that you need to remember. Whether it is 70S or 80S, whatever it is having the two subunits. It is having two subunits. There is one important information, so that I will link up with molecular basis of inheritance. So please remember all this. Whatever I am writing here, though not mentioned in cell chapter, it is important for examination. So please remember here. 70S ribosome and then 80S ribosome. For the 70S ribosome, what is the larger subunit? 50S is larger and smaller is 30S. But for 80S, 60S is larger and 40S subunit is smaller. Up to this it is nice. In the case of 50S, what are the rRNAs that are present? rRNAs that are present are 23S rRNA, 23S rRNA and 5S rRNA. 23S rRNA and 23S rRNA and 5S rRNA. But in the case of 60S, 28S rRNA, sorry, 5.8S rRNA and 5S rRNA and 5S rRNA. Please observe the relation here. Here two types of rRNAs, here three types of rRNAs, and 30S rRNA is having only one, that is 16S rRNA, and here 40S is also having only one, but it is 18S rRNA. So observe the relation here. In the in the molecular basis of inheritance of class 12th, they have mentioned all these names. They have mentioned all these names, so please remember this. Next one, under the translation, they have mentioned about this particular one. So please observe all these things. So better please remember at least the list of these rRNA molecules. Along with rRNAs, some proteins are there, some proteins are there, some proteins are there. So in this way, RNA plus proteins, finally they are forming subunits. When subunits are combined, they will form ribosomes. So each ribosome is having two subunits, one is larger, other one is smaller. This is the list. And out of this list also, what is important for examination? 23S rRNA of 50S subunit present in 60S ribosome and 28S rRNA of 60S ribosome subunit present in 80S ribosome. So these two, 23 and 28. These two are having enzymatic activity. This was mentioned in translation of molecular basis of inheritance. So please remember this. Okay, these two, 23S rRNA and 28S rRNA. These two are acting as enzymes. What is the name of enzyme activity? Peptidyl transferase. In the regular lecture, I have very clearly explained all these points. So sorry. So these two are the rRNAs. Sorry. These two are the rRNAs, ribosomal RNAs that are acting as enzymes. If RNA acts as enzyme, it is called as ribosome. So these are example under ribosome, and these are the rRNAs having enzymatic activity, and the name of enzyme activity is peptidyl transferase, that is involving in protein synthesis, translation, everything. So please observe this. These are very, very important points. So please remember this. And after this, next one is regarding the S. What the S value indicates? S value is nothing but Swedberg unit. Actually, it is the sedimentation coefficient. Indirectly, it will tell us what is the density, what is the size of this one, size and density of particular subunit. Clear? Anyhow, for this calculation, actually it is the time calculation, and that calculation is not needed at this level. So simply remember like this. Clear? Next one. Regarding the cytoskeleton. Cyto means a cell. Skeleton is the support. Actually, for our body, the bones and cartilage, they are acting as the skeleton. They are providing support for the body. In the same way, inside the cell, inside the cell, there are some structures that are providing support to the cell. So the structures that are providing support to the cell, so they are called as cytoskeleton. And what are the important points? These are network-like. They are interconnected network-like. Filamentous means very thin thread-like structures, and proteinaceous. All these are proteins only. All these are proteins only. Very, very important. Proteinaceous word is very important for examination. Structures. And what are these three types of proteins? Microtubules, microfilaments, and intermediate filaments. These are the three types of proteins that are acting as cytoskeleton. And what is the overall function of the cytoskeleton? Helping in mechanical support. They are providing support to the cell, and they are also helping in motility, mainly in the case of pseudopodia concept, if you know. So there you can understand that it is helping in the movement of the cell also, and then it is also helping in maintenance of the shape of the cell. If you observe, wherever cell wall is there, their cell wall will decide the shape. But for the animal cell, if you take, cell wall is not there, but still you will say that so and so cell is having so and so shape. Now who will provide the shape? So please remember, wherever cell wall is not present, there who will provide the shape? Cytoskeleton will provide the shape. Clear? Even plant cells also have the cytoskeleton, but still shape will be decided by outermost cell wall there. Okay, so please remember, there it will provide support, but shape will be decided by cell wall. Next one is regarding the cilia and the flagella. So cilia and flagella, these are the surface structures that we are observing in animals, that we are observing in the eukaryotic cells in general. Let us see. But please remember, the term flagella we studied even in prokaryotes also, but we have never studied cilia in the prokaryotes. Please remember, cilia are never present in prokaryotes. They are present only in the eukaryotic cells. Observe carefully. And why we are studying these two together? Because structurally these two are 100% exactly same. Structurally 100% exactly same, but the length wise there is a small difference. That's it. Length wise there is a difference, but structure wise 100% they are exactly same. That's why we are studying them together. Okay, and please remember, they are hair like outgrowths of the cell membrane, plasma membrane. They are actually starting from the plasma membrane. Anyhow, under the structure I will tell you. First, let us see about the cilia. Length wise, if you see, cilia are shorter when compared with the flagella. And how they are working? They are working like the wars. See, they are pushing the surrounding world, surrounding fluid, yes or no? So that they are causing the movement of surrounding fluid, and sometimes, I am not saying always, sometimes they can cause a movement of the cell also. That's why under the locomotor structures we are studying the cilia. Cilia can help in movement of the cell. If you are talking about the paramecium, how it is able to move? With the help of cilia only. But if you are talking about the human, we are also having the cilia. So in this respiratory tract, we are having ciliated cells. Cilia containing cells are present. And if you are talking about the fallopian tube of human female reproductive system, there also ciliated cells are present. Their cilia are present. There also cilia are moving. But cilia do not cause movement of the cell in those conditions. Yes or no? So that's why all the ciliated cells cannot move. So in some cases, ciliated cells, they are moving, their cell is moving, and in some cases they are causing movement of some mucus or some fluid or some dust or ovum or something, they are causing the movement. Clear? So this is what the function of this particular cilia. Now we will go with the flagella. Flagella, comparatively it is longer with respect to that cilia, and here generally, please remember, here also not always, here also there is some exception. So here generally it is causing the movement of the particular cell. So generally flagellated cells can move, but under the animal kingdom, under the sponges, phylum Porifera, we studied about coinocytes. Coinocytes are having flagella, but they cannot move. So please remember this. So that's why most of the flagellated cells can move. This is what we are saying. Now we will talk specifically with respect to the human. In the case of human, if you are talking, we have ciliated cells and we have flagellated cells also. So now observe carefully. If you are talking specifically for human female has ciliated cells. Human female has ciliated cells, but flagellated cells are not present in human female. But human male, if you are talking, human male has both ciliated cells and flagellated cells. In the case of human, the only type of flagellated cells are sperm cells. That's it. The only type of flagellated cells are sperm cells. So please remember this. This is one thing. Next one, regarding the cross-section structure, if we are observing 9+2 microtubular arrangement we are observing here. Clear? Before directly going with this structure, first let me explain how exactly the arrangement is present. Suppose this is the plasma membrane. This is the plasma membrane. Now we are drawing the cilia or flagella. Cilia will be shorter, flagellum will be longer. That's it. Okay. This is the plasma membrane. Please remember, plasma membrane itself is having fold here. Okay. And now what we are saying here, at the level of plasma membrane, there is a basal plate, and below this there is a basal body. Basal body is present, and this basal body is having 9+0 arrangement like centriole. Like centriole, it is having 9+0. And this upper side, this part is called as core or axonium of this particular flagellum or the cilium. Okay, and here it is having this structure, and this is 9+2 arrangement. Core or axonium is having 9+2 arrangement, and you can call this particular one as a shaft of cilium or flagellum. Clear? These are the different things that are present. Basal body, once again I am repeating, basal body is having centriole like 9+0 arrangement, whereas the shaft or core or axonium is having 9+2 arrangement for both cilia and flagella. This structure is same for both cilia and flagella. Only difference is cilium is shorter, flagellum is larger. That's it. Clear? And now, anyhow, this 9+0 under the heading of centriole, or centrosome we will study. So now we will focus on this 9+2 arrangement only. So please remember, if they are specifically asking basal body of cilium or flagellum, 9+0, core or axonium of cilium or flagellum, 9+2. If they are not mentioning anything, so which of the following type of microtubular arrangement is seen in cilium or flagellum? Simply they said, they have not mentioned which one specifically, and you need to select one. Which one you need to select? 9+2 only you need to select. Understood? So now we will go with that 9+2 microtubular arrangement. So here 9 peripheral doublets are present. 2, 2, 2, 2 are present. Likewise 9 are there. 9 peripheral doublets are present, and adjacent 9 peripheral doublets are connected by linkers. And these linkers are also called as interdoublet bridges. These are present on outer side. Now at the center, 2 singles are present, and these are called as a singlet microtubules, and these singlet microtubules are connected by again some bridges, and around these singlet microtubules, this red one, so there is a central sheath. Yes or no? Next one, from the peripheral doublets there are some protein strands, they are coming and attaching to this central sheath, and they are called as radial spokes. So since 9 peripheral doublets are there, in the cross section, how many radial spokes we can see? 9 radial spokes we can see in the cross section. Okay, one from each of this peripheral doublet. And along with this particular one, even the not mentioned NCRT, they have observed the diagram here, 1, 2, 3, 4, 5. Likewise some arms they have mentioned, they are called as a dynein arms, dynein, dynein arms. These are called as dynein arms. So this is what we are actually observing in the cross section. And if you see on the outermost what is present, this actually green, it has actually came inside, but actually it should come and stop at the green line. And actually what is this? Plasma membrane. So why they are talking about plasma membrane here? Because if you are observing the cross section, so what you will get here? Plasma membrane only. So that's why on the outer side, edge will be what? Outer that circle will be plasma membrane. So this is how exactly the structure of the cilium or flagellum is present. Clear? So these are the points that we already discussed. So go through it. Next one is regarding centrosome or the centriole. So please remember, centrosome, one centrosome has 2 centrioles. Please observe carefully. Now it is completely called as one centrosome. Inside this one centrosome, 2 centrioles are present, and along with this some pericentriolar material, PCM, pericentriolar material. These names were very clearly given in NCRT. Observe carefully. 2 centrioles plus pericentriolar material. Combinedly it is called as one centrosome. Some students are not getting difference between what is centrosome and what is centriole. So one centrosome is equal to 2 centrioles plus pericentriolar material. This is what about the centrosome. So now anyhow, now we can focus on one single one. But before going with this, what is the angle between these two? They are present perpendicular to each other, 90 degrees to each other. And now if you are observing any one of these, and if you are observing the cross section, and here we are observing 9+0 array. For the cilia, flagellum, shaft or core or axonium, we are observing 9+2 array. Array means arrangement, but here we are observing 9+0. Even in the basal body of cilia, flagella also we are observing 9+0 only. So what is this 9+0? 9+0 in the cross section, what we are observing? 9 peripheral we are observing. There also 9 peripheral. But in cilia flagella, 9 peripheral are doublets, only 2 microtubules are present. But here how many are there? 3 microtubules are present in each fibril. Each fibril has 3 microtubules. That's why in the cilia flagella 9 doublets are there, but here 9 triplets are there. And there 9 doublets are connected by linkers or inter-doublet bridge. Here triplets are connected by linkers or inter-triplet bridges. This is the difference. But in the cilia flagella at the center 2 singlets are there. But here at the center there are no microtubules. So at the center there is a protein mass, it is called as hub. And directly from the hub, these peripheral triplets are connected by radial spokes. These are also protein strands. So her central hub, and through the radial spokes, it is connecting to peripheral triplets. At this level, they have given only this information, but X, Y, and some other things are also there, but they are not needed for examination. So leave it. This is how it is connected. So now overall, if you are observing in the 9 plus 2, 9 outer, 2 center. 9 outer are again doublets. So 9 into 2, 18 plus at the center 2 are there. So that's why total 20 microtubules are present. Yes or no? In the cilia flagella cross section, if you are observing for the axonium, so let me do the calculation part. So for cilia and flagella, if you are observing 9 plus 2, and these 9 are again 2. Yes or no? They are doublets. So 9 into 2, 18 plus these 2 are singlets. So 2 into 1. So 2. So total how many microtubules are present? So here total 20 microtubules we are observing in the cilia or flagella. But for the centriole, if you are observing, 9 plus 0, anyhow it is 0. So this 9 are again, each one is having 3 microtubules. So that's why it will be 27 microtubules you are observing. But radial spokes, here also 9, here also 9. So comparatively you just observe it so that it will be easy for you. So these are the information that they have given. So centrioles, so they form the basal body of cilia flagella. So basal body of cilia and flagella is also having 9 plus 0 arrangement. And next one, spindle fibers. So please remember, in the case of animal cells, centrosomes are present. They are helping in formation of the spindle fibers during the cell division. And here also I wanted to tell you one more very important point. That's where generally students are making mistake. Suppose this is an animal cell. Suppose these are the spindle poles. Centrosome will become the spindle pole, yes or no? And inside this, 2 centrioles present perpendicular to each other. They are present like this. And during the cell division, what we say? Spindle fibers will arise from this spindle pole. But if you are clearly observing this, from where exactly you are writing spindle fibers? From the outer side only you are drawing the spindle fibers. From the outer side only you are drawing the spindle fibers. Please observe this carefully. You are not touching the spindle fibers to these centrioles. You are not touching these spindle fibers to these centrioles. So please remember this. That means from where exactly they are coming. So around this, in this pericentriolar material, there are some satellites. Satellites are present. And that there are also some group of proteins, and actually they are important for this particular spindle fiber formation. So that is the reason why, anyhow, once again in cell cycle also I will tell you. So that's why even though these centrioles are not present, in the case of plant cell, still spindle fibers can be formed, because centrioles, directly those 2 centrioles are not involving in spindle fiber formation. This pericentriolar material is very important for spindle fiber formation during cell division. That's why in the case of higher plants, centrioles are not present, but still during cell division, spindle fibers will be formed. Spindle poles will be formed, because some proteins are required. Those proteins will come at that time and they will help in the formation of this particular structure. That's it. So that's why please remember, whenever you are drawing these spindle fibers, don't attach these directly to these to centrioles. Don't do this mistake. This is a mistake. This is a wrong concept. So this is the correct one. Okay. So understand the point. That much is enough. And remaining things we can see in the cell cycle. Now let us move further. And the last concept here, we are going through nucleus, and this was discovered by Robert Brown, and the name chromatin was given by the Fleming, because when we are using some basic dyes, please understand why basic dyes. Why we need to use basic dyes? Why not acidic dyes? Why basic dyes? Because the DNA is already negatively charged. DNA is already negatively charged. So basic stains, basic dyes or basic stains can go and bind with DNA so that you can see some color. So that's why when stained with basic dyes, we can see some color. So colored threads, so chromatini, chroma is color, tini is the thread. So colored threads were observed by this scientist and he gave the name called chromatin for these threads present inside the nucleus. So name was given by Fleming. Please understand each and every logical word you need to understand. Next one, in the case of interphase nucleus, we are observing some important information. What we are observing? Nuclear membrane we are observing, or nuclear envelope you are observing, and the nucleoplasm, also called as nuclear matrix, we are observing, and the nucleolus we are observing, and along with this chromatin we are observing. So these are the four important components that we are observing. First we will talk about the nuclear membrane. So nuclear envelope is having two membranes, outer membrane, inner membrane. Yes or no? So because of this nuclear envelope, so content of the nucleus is called nucleoplasm. Now this nucleoplasm is separated from cytoplasm due to presence of nuclear membrane. But though it is separated, now here the nuclear membrane at some locations, outer and inner membranes, they are fusing together so that they are forming some nuclear pores. Through the nuclear pores, some materials from the cytoplasm can enter into nucleoplasm. Some materials from the nucleoplasm can enter into cytoplasm. So that's why these nuclear pores are acting as gates between nucleoplasm and cytoplasm, so that proteins and some molecules can easily pass between these things. Clear? This is one important information we need to understand. Next one. So these are the two parallel membranes, and the gap between these two membranes is called perinuclear space, or in general you can say intermembranal space also, no problem. And in NCERT they gave some range between 10 to 50 nanometer is the size that they have mentioned for perinuclear space they have mentioned. Clear? Next one. The outer membrane of nucleus is continuous with endoplasmic reticulum, and even outer membrane of nucleus is also having ribosomes. Very, very important point. So please understand this particular one. Clear? Next one. How many nuclei are present? Generally one nucleus is present, but for this also exceptions are present. In some cases, single cell can have more than one nuclei, and they are called as multinucleate cells. Generally we are observing in the fungal cells, but even in human also, if you are observing, skeletal muscle cells are multinucleated cells. They are formed due to fusion of the cells. So that's why multinucleate condition we are observing in human skeletal cell. Whereas some cells, they don't have the nucleus. When they are matured, they are losing the nucleus. They are called as enucleated cells. Enucleated cells. One important example is majority of the mammals, RBC of mature RBC of majority of the mammals. So human mature RBC do not have the nucleus. So these are called as enucleated cells. But even though nucleus is not present, still we are considering them as living cells, because still living reactions are taking place. They are involving in so many functions. Next one. So with this, nuclear membrane concept is completed, and then we are going with nuclear matrix. It is also called as nucleoplasm. The semi-solid dense matrix, or the fluid that is present inside the nucleus, is called as nucleoplasm. That's it. And inside this we are observing another spherical shaped non-membrane bound structure, generally one in number, and depending on the cell function sometimes more also can be present, and this is called as nucleolus. Singular is called nucleolus, plural is called as nucleoli. So this is what they are talking. And what is the function of this one? So this chromatin is passing through the nucleolus. So when chromatin is passing through the nucleolus, within the nucleolus some genes are present. So mainly the genes related to rRNA synthesis, ribosomal RNA synthesis are present here. So since ribosomal RNA synthesis genes are present here, ribosomal RNA is synthesized here. Now proteins from cytoplasm will come here. rRNA and proteins will combine and they will form ribosomal subunits. And subunits will go again to the cytoplasm. And in the cytoplasm, when subunits are combined, they will form ribosomes, and then they are involving in protein synthesis. That's why protein synthesis and nucleolus size and number, they have given a relation in NCERT. A cell that is actively involving in protein synthesis will have large nucleoli, large and more numerous, more number of nucleoli. Now what is the relation here? What is the relation between protein synthesis and large size and more number of nucleoli? What is the relation? The relation is For protein synthesis, ribosomes are required. For ribosomes, ribosomal subunits are required. For ribosomal subunits, rRNA is required. And who will synthesize rRNA? Nucleoli. If more protein required means more rRNA is required. If more rRNA is required, large size nucleolus and more number of nucleoli are required. This is what the relation that they have mentioned in NCERT. Observe each and every sentence and have the relation. Then you can understand all these things. And the last one is, in the interphase nucleus, chromatin is present. Please remember, interphase means G1, yes, G2 phase. In these phases, we are using a term called as a chromatin. Whereas during the cell division phase, M phase, because of the condensation, that chromatin, now it is called as a chromosomes. Words were very clearly used, so please observe carefully. Clear? And next one, and this chromatin or chromosome, it is having DNA and some basic proteins. Histone proteins are basic proteins, right? So some basic proteins called histones are present, and some non-histone proteins are present, and along with this even RNA is also present, and many other things are present within this chromatin or chromosome. And then calculation. So 46 chromosomes combinedly, they are having 6.6 into 10 to the power 9 base pairs. In the molecular base of inheritance, we have studied about this value. And then when you are multiplying with distance between two base pairs, now what is the length you are getting? In MBA, in molecular base of inheritance, we have done this clear calculation. What is the value we got? 2.244 meters. I told very clearly, 2.244 meters you are getting for 46 chromosomes. So for the 2.244, so they have made it as approximation as 2 meter. So in single cell, please remember, in single cell of human, 46 chromosomes are there. If you are arranging all the 46 back to back, what is the length you are getting? Approximately 2 meters. This is what the information is. The DNA present in one cell is having approximately 2 meter length. This is what the information they are saying. But in NCERT, what they said? Approximately 2 meter length of DNA is distributed into 46 chromosomes. What is the meaning of it? If you are attaching all the 46, you will get this 2 meter length of DNA. This is what the meaning is. And then regarding the structure of chromosome. So chromosome is a condensed form, and generally in the metaphase of mitosis, we are isolating the chromosome for the morphology study, so that we can easily study the chromosomes. Now here the ends of chromosomes are called as telomeres, and generally there is a constriction. There is one compulsory constriction. There is one compulsory constriction. That's a compulsory constriction is called as a primary constriction. It is also called as centromere. And it may be present at the center. It may not be present at the center of chromosome. If it is not present at the center of chromosome, then we will see one short arm and one long arm. We will see one short arm and one long arm. Short arm is indicated with P and long arm is indicated with Q. Okay. So now this is what, for our understanding purpose, generally we are studying like this, but during the metaphase, generally we are observing the dyad chromosome with two chromatids. So this is how it is present, but both the sister chromatids are attached at single centromere. So this is how we are talking about this particular structure of the chromatid. So we need to know about centromere. It is also called as a primary constriction, and the kinetochores. What are the kinetochores? There are some protein plates that are present on either side of centromere. For one centromere, two kinetochores are present. So this is what the calculation is. And arms. So from the centromere, the segments, the length of this chromosome that is present on either side are called as arms, and the ends of these arms are called as telomeres. These are the important words that we need to understand. Next one is regarding the secondary constriction. What is the secondary constriction? So please remember, so don't think that only for metacentric chromosome, it can be present on anyone. So here what happens, for some of the chromosomes, anyhow, for all the chromosomes, centromere is compulsory. Along with this, some chromosomes have another constriction. That another constriction is called as a secondary constriction. It is also called as nucleolar organizing region, NOR, nucleolar organizing region. In the regular class, I have given some information regarding this in relation with that nucleolus, protein synthesis and all the concepts. Okay, go through that. So it is also called as nucleolar organizing region. Please remember, this secondary constriction may present on metacentric, submetacentric, acrocentric, any of these. Okay, even on telocentric also it can be present. In different organisms, in different chromosomes it may be present. Please remember here. But some students, they are thinking that in NCERT, for our understanding purpose, on one chromosome they have mentioned, and by chance it is a metacentric chromosome. So many students, they are thinking that secondary construction is present only on a metacentric. They are thinking like this. That is a wrong thing. That is a wrong interpretation. So don't think like this. It can present on any of the chromosomes. Clear? This is what the information is. Next one, types of chromosomes based on position of centromere. If centromere is present at the center of chromosome, two equal arms will be produced. So that since it is present at the center, it is called as metacentric chromosome, and it is also called as isobrachial chromosome. Iso means equal, brachial means arms. Isobrachial, equal arms chromosome. So which of the following is the isobrachial chromosome? It is the metacentric chromosome. Clear? And during anaphase, what is the shape that we are observing? V shape we can observe here. Next one is submetacentric. Centromere is present slightly away from the center, so that one is long arm, other one is short arm. During the anaphase, we are observing L shape for this. Next one, acrocentric. So here centromere is present close to one end, that means nearer to one end. So that one is very short arm and other one is very short arm, other one is very long arm. And here we are observing J shape during the anaphase. And the last one is telocentric. In the telocentric, telo means what? Ending, right? So at one end centromere is present. So that's why here we are observing only one arm. But please observe NCRT diagram. In the NCRT diagram, for the telocentric also they have mentioned one small short arm, and they have even labeled it as short arm also. Why they have mentioned it like this? Is there any mistake in that? There is no mistake. Please remember, whatever the general magnification, when we are observing chromosomes at a particular magnification, we are observing under microscope. At that magnification, you cannot see that small short arm, some cases. And if you are going for further higher magnification, you may see that short arm. So even the very short arm is present, and still it is considered as a telocentric, because at the normal magnification we cannot notice that very short arm. But in this case, at the normal magnification, you can notice the short arm. That's why it is considered as acrocentric. But this is considered as telocentric. So please observe NCRT diagram also, and I gave the proper explanation in the regular lecture for this. Clear? Next one, regarding the microbodies, information was not given much. Simply they said that many membrane bound minute vesicles they have given. Many students, what they are doing is they are reading that many membrane bound. How many membranes are present for microbodies? If we are asking, they are saying that many membranes are present. That is wrong. That is wrong. You should not read like this. Many single membrane bound. You please add in the NCRT. You please add, after many you just add single. There are many vesicles are present. The meaning is many vesicles are present. And what type of vesicles are those? Single membrane bound vesicles. So in this way, all the microbodies, they are single membrane bound only. So please remember this. So many microbodies are present. That's what their meaning is. This many word you should not attach for membranes. So there are many vesicles. Minute means small vesicles are present, called microbodies, are present in both plant and animal cells, and they are having various enzymes, so that they are involving in various functions. But all these microbodies are single membrane bound. So that's why under the list of double membrane bound, single membrane bound, so I wrote microbodies under single membrane bound. So please observe this carefully. So this is what one important point, and these are present in both plant and animal cells. Clear? But at least in the NCRT they have mentioned about the peroxisomes. So peroxisomes, they have at least very clearly given the labeling in both plant and animal cells. So observe that carefully. So what actually they are doing? So generally they are having the catalase enzyme, so that H2O2 is converted into H2O and O2. This is one function. And not only this, peroxisomes generally involving in fat oxidation, breakdown of the fat. Okay, so this is one major important function. In the case of plants, these peroxisomes can also be called as glyoxysomes, and along with the fat breakdown, so they can also involving in glyoxylate pathway also in the case of plants specifically. Okay, so this is some important information. So this much is enough, and then finally we will go with some important difference between prokaryotes and eukaryotes. If you are observing with respect to nucleus, prokaryotes do not have nucleus and eukaryotes have well defined nucleus. Organelles, membrane bound organelles are present in eukaryotes, not present in prokaryotes. But only type of organelles present in both prokaryotes and eukaryotes are ribosomes, which are non-membrane bound organelles. Regarding the DNA, so in the case of both prokaryotes and eukaryotes, DNA is the genetic material, but in prokaryotes, DNA is a naked DNA. It is not associated with histone proteins. Please remember here, some students are making mistake here. Why prokaryote DNA is naked? So suppose I am giving an assertion, prokaryote DNA is naked. What is the correct reason that you are giving? Some students are giving wrong answer. Some students are saying that it is not covered with nuclear membrane. That's why it is naked. That is wrong answer. It is not with respect to nuclear membrane. Why prokaryotic DNA is called as naked? Because histone proteins are not present. That is the correct answer. Don't write about nuclear membrane. So in the case of prokaryotes, histone proteins are not present. That's why it is naked DNA. But in the case of eukaryotes, it is not naked DNA because histone proteins are present. That's why in prokaryotes, DNA is present as chromosome. In eukaryotes, DNA is associated with histone proteins. That's why it is present as chromosomes. But in prokaryotes, it is not present as chromosomes. Next one, flagellum. Flagellum is present in both prokaryotes and eukaryotes, but structurally, chemically, it is completely different. Prokaryotic flagellum is made up of flagellin protein, but in the case of eukaryotic flagellum, mainly it is made up of tubulin protein because microtubules are present. Yes or no? Next one, mesosomes. They are characteristic for prokaryotes. We are not observing in eukaryotes. Inclusion bodies. Technically, if you observe in higher classes perspective, if you say inclusion bodies are present in both prokaryotes and eukaryotes. But as per NCRT, they are present in prokaryotes. They have not mentioned in eukaryotes, but even in eukaryotes also they are present. But the type of inclusion bodies, it depends on the organism to organism. Clear? With this, this particular part is completed. With this, the revision of cell chapter is completed. Clear? So in the next lecture, we can go with some other chapter. So once again, I am telling you, the information regarding these revision lectures will be available on app and also YouTube, but the test series is available only on the app. Clear? So with this lecture is completed. We will meet in the next lecture. Thank you.